Okay, well, thank you everybody for coming. Thank you for coming on time, and a really warm welcome to our Movement Disorder Centre Symposium. I'm just going to give you a very brief introduction to who we are and what we do. So at UCL and at Queen Square, about 8 years ago now, we set up, this is who we are. So the NIHR have set up these biomedical research centres across the UK, and the UCLH Biomedical Research Centre allowed us to form the Movement Disorders Centre here at Queen Square. And really it's a group of interested, engaged people who really seek to try to improve the lives of people with rare movement disorders and common movement disorders. So this is my wonderful board at the top, and they've really been unbelievably supportive and helpful in trying to launch a centre that can unify all of our efforts into translation. And then, of course, the team is much wider. These are just the pictures I've managed to grab, but actually we're a large group of up to 30 principal investigators and clinicians and clinical academics who are all working in this space. And we have a lot of early career researchers as well, so huge thanks. And just before I start, also a big call out to Hannah, who's our coordinator, who's helped to organise the day. Thank you, Hannah. And Angelina, our senior research nurse, who supports all of the trials and work that we do in the centre. So what do we do? Well, when we did set up, our vision was really about accelerating translation. It was this idea that it takes so long to go from bench to bedside, 10 to 15 years. And in movement disorders especially, but especially the degenerative and neurodegenerative movement disorders, we actually have made very little progress in this concept of disease modification. So we need to kind of accelerate the translational process, but we also need to reach therapies that are actually going to have benefit for people. So this idea of embedding precision medicine, which was a really ambitious goal back in 2016, 2017, has really driven us to come together to have all of our patients through our trusts, through the universities, on one common platform that everybody can then use for research. We have many research cohorts across the centre. We have a lot of preclinical science. You're going to read about it in your brochure, which I hope you can enjoy, about many of the latest developments that have happened in our centre. And then, of course, we run many trials that span experimental medicine through to phase 1, phase 2, and indeed phase 3. And some of those breakthroughs have really been in the last 2 years since we last ran this symposium. So I hope you'll see that progress is moving eventually towards this final goal of precision medicine. But actually, it turns out along the way that to reach precision medicine, what we need is precision biology. And so much of what you're all doing in the room is really pinning down mechanisms, trying to get with precision to what mechanism affects which person, ultimately to design drugs that we can treat the right person at the right stage in their condition, and ultimately improve the outlook of therapies. Okay, and then it just really leaves me to thank all the funders and the sponsors and all the people who've made today possible. We've put together a programme. We hope it's a really exciting programme that showcases what's happened at UCL, but it also showcases sort of UK PLC, where we are more broadly in the UK environment. And so you'll hear talks from many people across the UK as well. And that's really been enabled because our partners have given us sponsorship and funding and taken us that step closer towards our goals. So a huge thank you to our funders. Please talk to the sponsors as well and interact with them in the breaks. And please do stay for the networking sessions as well, because I hope they'll be very fruitful and productive. And with that, we'll move on to the first session. Okay, all right. So we're going to start with, each session is sort of themed. The first session is themed on pathomechanisms in movement disorders. And of course, I'm very pleased to invite Professor Schapira to give his talk. I don't think Prof. Schapira needs any introduction, so I'm not going to read out your bio, Tony. You'll be pleased to know, but you can read about him in the brochure. And really, Tony's work has spanned, obviously, a long time. It's been hugely impactful, and for us, it beyond his science, he's really helped us generate this ecosystem here at UCL in which we've been able to thrive. So, Tony, I'm really pleased to invite you to give your talk.
Thank you very much, Sonia. And thank you to the MDC for the invitation to talk to you today on GBA1. So it's an update on GBA1. glucocerebrosidase. And I'm going to try and cover the whole spectrum of the relevance of GBA1, specifically in Parkinson's disease, but I'll also be mentioning one or two other disorders as well. And I thought I would start at the end with the takeaways. So I think it's helpful if you leave this session and this particular lecture with some specifics about Parkinson's disease and GBA1 if you're not familiar. So importantly, it's the commonest known genetic association with Parkinson's disease by far. In the UK, 12% of our Parkinson patients have GBA1 variants. So it is, as I say, by far the commonest genetic association. But if you flip that and ask how many people with a GBA1 variant get Parkinson's disease, the answer is only 10 to 15% by age 80. So the penetrance of GBA1 is relatively low, despite the fact it's the commonest gene variant in Parkinson's disease. Interestingly, however, there's a connection between the genetic GBA1 association with PD and idiopathic non-GBA Parkinson's, because in the Parkinson brain, specifically in the substantia nigra, there's a significant reduction in glucocerebrosidase activity, the enzyme encoded by GBA1. So there's this connection between the genetic form and the idiopathic form through this pathway. Clinically, GBA1 Parkinson's disease, that's just been people with Parkinson's disease who have a variant, are indistinguishable from those who don't have a variant or idiopathic Parkinson's disease. So on a group-to-group analysis, there are some differences, but mainly clinically, by imaging, therapeutic response, and pathology, there's very little difference. However, some of the features of Parkinson's disease are exacerbated by what we call the more severe variants of GBA1. And these severe variants result in earlier onset of Parkinson's disease, more rapid progression, more cognitive dysfunction, more non-motor problems. Not all GBA1 variants are the same, and I'll come back to that. How do GBA1 variants result in Parkinson's disease? We don't know, but there are many candidates, and they're not mutually exclusive. So there are several pathways here that may be involved downstream of the effect of a GBA1 variant. And over the past few years, identification of this pathway and its relevance to Parkinson's disease has resulted in a significant therapeutic focus on GBA1, such that there are numerous trials currently in phase 1, phase 2, and phase 3 targeting the GBA1 pathway. So those are, if you will, the takeaways from GBA1. And over the next few minutes, I'll just try and elaborate a little bit more on some of those aspects. But before I do so, I just wanted to highlight a thank you to all those people in the GBA1 team that have done all the hard work that I'm going to reference that has come from Queen Square. So thank you to all of these people. I'm going to start with the gene. The gene is relatively small, it's 7.6 kilobases, but it is very interesting because it has a pseudogene almost next door on chromosome 1. And there is a 95% homology between the pseudogene and the gene. And in some areas, particularly exons 8 to 11, the homology rises to about 98, 99%. So it's almost an identical gene, pseudogene, non-coding, although mRNA is produced from that pseudogene. The point is that this makes sequencing of GBA1 variants extremely complex. So it's not the sort of thing that actually you get accurately back from NeuroBooster. And even on other platforms, the sensitivity and specificity is low. In order to get up to about 99% plus accuracy, you have to do long-read sequencing on nanopore, followed up by Sanger. So there are over 370 different variants, and they're distributed throughout the exons. But as you'll see, the variants are not all the same, as I've implied. Some are mild, some are severe, and some are what we call risk variants, and I'll come on to that in a second. And you see the distribution of those mainly between exons 8 and 11. Now, the distinction between the different variants is defined by what effect these have on Gaucher disease. Gaucher disease is the presence of two abnormal GBA1 alleles. It's an autosomal recessive disorder. Now, I'll tell you a little bit more about that in a moment. So severe variants are associated with what's called type 2 and type 3 neuronopathic Gaucher disease. Mild variants are associated with Gaucher disease that's non-neuropathic or type 1. And risk variants, when present in homozygous or biallelic form, do not cause Gaucher disease. So that's the definition. So there are, as I say, over 370. About 5.9%, 6% of those are severe. About 22% are severe, 6% are mild, and the vast majority are of unknown, very small number of identified risk variants. So severe variants are such as the L444P, more common in Asian communities. The milder, the N370S, more common in European and Ashkenazi. And risk variants are present all over, and there are different types of risk variant that you can see. The most recent one just has come out last month. So this is the gene. The protein plays an extremely important role in biochemistry. It's part of the ceramide pathway, the glucose-finger-lipid pathway, ceramide pathway. It's a relatively small protein, 536 amino acids. Molecular mass is only 60 to 65 kilodalton, depending on post-translational modifications. But it plays a hugely important role in a pathway that is littered with neurological disorders. The diseases. So mutations or variants of the GBA1 gene are associated with several diseases, Gaucher disease to begin with. Philippe Gaucher was a dermatologist who identified a young female with hepatosplenomegaly, and the cause, of course, at that time was unknown, but this association bore his name. And it wasn't until Roscoe Brady in the 1970s who identified the enzyme defect in what was known as Gaucher disease as glucose ribosydase. Gaucher disease can present at any time of life, from infancy to late adulthood. I've mentioned type 1, which is generally mild, hepatosplenomegaly, joint problems, anemia, and neuronopathic, which has severe brain involvement. So this is Gaucher disease, autosomal recessive biallelic GBA mutations. Ari Zimran, a neurologist, metabolic neurologist in Israel, was the first to find the association, or to report the association between Gaucher disease and Parkinson's disease, because he saw a number of his patients with Gaucher disease developing Parkinson's disease. And interestingly, a number of the relatives who are obligate carriers developed Parkinson's disease. And he was the first to report this association. But in fact, it was Ellen Sudransky who really made the connection and did this meta-analysis of the genes associated with GBA1 and confirmed the association with Parkinson's disease. There is a small dose effect. So here what we're looking at is a GBA1 variant that can cause or be associated with Parkinson's disease either in homozygous or heterozygous carrier form. So just the presence of one variant increases your risk for Parkinson's disease, but of course does not cause Gaucher disease. Those with Gaucher disease who get Parkinson's disease tend to do so slightly earlier than those who are the heterozygote carriers, but it's not a major difference. I mentioned that the penetrance is relatively low, so only about 10 to 15% develop Parkinson's disease if they carry a gene, but it's the commonest gene association. And it's worldwide. So you can see it in a significant proportion of Chinese Parkinson's disease, Ashkenazi, and significantly in those of West African descent. The relevance of the severity of the gene mutation is important in counselling. And this is counselling not only the patient, but also the family. Because severe gene mutations, as I mentioned, like the L444P, have a significantly increased odds ratio of developing Parkinson's disease than the others. So if you carry a severe mutation in one allele, your risk of getting Parkinson's disease is substantially increased, somewhere between 6 and 30-fold. A mild variant is much less frequently associated, so obviously a fewer proportion of those that carry the mild variant get Parkinson's disease. And then the risk variants that are the commonest have the lowest risk of getting Parkinson's disease. So you can see the type of variant and what the person carries is significant in terms of their risk of developing the disorder. What are the clinical features of GBA1 Parkinson's disease? Well, earlier onset, on a group-to-group analysis, more cognitive impairment and faster progression. And you can see this in this graph, actually, from a paper a little while back. But you can see that the accumulation of cognitive deficit to dementia varies according to whether you carry a GBA gene, and here these are non-carriers. If you carry a mild GBA gene or a severe one. So you can see carrying the severe variant significantly increases your risk of cognitive impairment. The pathology is the same. It's typical Lewy body pathology, but generally in GBA1-associated PD, particularly severe mutations, there's a much higher Lewy body load. The imaging is identical. Interestingly, in pre-symptomatic carriers of GBA1 genes, you can see microglial activation in the brain before dopamine loss. The pharmacology is the same. The patients respond, they get dyskinesias. And response to DBS is the same. There was some concern from a paper that was published a few years ago that people who with GBA1 genes that were having DBS seemed to worsen more rapidly. But actually, more careful analysis of that data in more recent papers have shown that patients with GBA1 genes respond well to DBS, but the presence of a severe mutation determines the progression of the patient, not the DBS. So this is an important feature because you don't want to not use DBS in what is 12% of the population. So this is where counselling may become valuable in knowing what type of gene the patient has. So in summary, it's common in Parkinson's disease, low penetrance, different forms of variant type, earlier onset, more cognitive dysfunction, more non-motor problems, more alpha-synuclein, and you see in the SAA, the C-identification assay, a very high specificity and sensitivity in GBA1. And interestingly, there's increased spread of alpha-synuclein in GBA1 models. And Jabbari has recently shown that in GBA1 mutations, there's a faster time to threshold in the SAA assay. And that is associated and correlated with onset and progression to cognitive dysfunction. So, a couple of papers that might be of relevance. This is about the severity, how many of our assumptions about GBA1 are actually, when you look at it, driven by the severity of the mutation. And so it's important to understand what mutation a patient has. And this is a consensus guideline on counselling for patients and relatives with GBA1. So what are the mechanisms? What are the downstream effects? Well, they are, as I implied earlier, multiple and not mutually exclusive. So glucocerebrosidase is a lysosomal enzyme, but it passes through the ER first, and mutant forms can get trapped and cause ER-associated stress. Mutated glucocerebrosidase can impair chaperone-mediated autophagy, which is important for alpha-synuclein. There's impairment of mitochondrial contact sites with lysosomes, and this can lead to mitochondrial dysfunction. Glucocerebrosidase has been associated with the maintenance and construction of mitochondrial complex 1 and is important for its function. And all of these effects lead to increased free radical-mediated damage and, importantly, the pathway to lipid dysfunction, because this is where glucocerebrosidase lies. So lipid abnormalities are common in cell models, animals, and now patients with GBA1 mutations. So what about the treatment of GBA1? Well, I've mentioned that symptomatic treatment is the same as for those who have idiopathic PD. But if you want to look at the specific therapy for GBA1, there are several different targets. There's the gene itself, and there's gene therapy for GBA1 currently in phase 1. There's attempts that using chaperones and HDAC inhibitors to increase the activity of glucocerebrosidase and improve its shuttling through the cell. Possibly enzyme replacement therapy. This is used to treat Gaucher disease, but it can't pass the blood-brain barrier. But new forms of ERT are now being developed. And finally, theoretically, substrate reduction therapy. So these are the current trials. So several have used ambroxol, cough linctus, very effective glucocerebrosidase chaperone. The first was a proof of principle. It gets into the brain, activates glucocerebrosidase. There's another one that looked at Parkinson's disease dementia, a relatively small study, not terribly well designed, but didn't show any difference. These are yet to be reported. It should come out later this year. And then there's a BL-GBA1 activator, chaperone type. Results should come out in Q3 of this year. There's the ongoing ambroxol phase 3 ASPRO-PD study. This is the largest study in GBA1. Currently recruiting, data will come out in 29. And there's the Norwegian ANEAT study on dementia with Lewy bodies. Substrate reduction therapy, so these are all targeting GBA. This is targeting substrate. That failed, so it didn't show any difference. And frankly, not a surprising result. So it wasn't targeting GBA, it was targeting the substrate reduction. And finally, gene therapy, which is still in phase 1. The future. What does the future hold? Well, obviously, we'll await the results of the clinical trials I've just mentioned. No doubt more will be coming on stream. But trying to find out who is going to get GBA1 Parkinson's disease. So this is an interesting study that shows the effect of the microbiome. And this study showed that there is a specific Parkinson's disease microbiome signature that is a composite of different species and a significant difference from control. But interestingly, the microbiome signature correlates with disease progression and severity of disease. But more importantly, that signature was identified in these people with GBA1 variants before they got Parkinson's disease. So these are asymptomatic carriers, but the ones in the dark are those who satisfy the MDS criteria for prodromal Parkinson's disease, olfactory loss, some RBD, etc. And all of those fell within the signature. So this suggests that maybe the signature can be used as a biomarker for those who are more likely to get Parkinson's disease. So this just a recent review that came out just a few days ago might be helpful in summarizing what I've been telling you. Important questions remain. So at the end of all of that, there's a lot we don't know. We don't know which of the carriers is going to get Parkinson's disease. We don't know if they can be predicted by biomarkers yet. We don't know why they develop Parkinson's disease, why some, not others, but also why, what are the downstream effects? And importantly, will GBA1 increasing glucocerebrosidase activity slow or prevent Parkinson's disease? And we'll know that only when you get the trial results. Thank you very much indeed.
Huge thank you, Tony. That was a tour de force over GBA1 and so much progress. So any questions? For...
Thank you very much. BL this morning just announced the failure of the phase 2B. I was wondering if you had any comments on the mechanism and the approach of allosteric modulators and chaperones, whether you see it as a mechanistic ceiling, and basically your view on this.
So you're one step ahead of me.
It was just this morning.
It's social media.
Okay.
Social media.
And what did it show?
No effect on the. They didn't release any detailed data. They failed to meet the endpoint.
Okay.
They'll release data later, but we don't have any detailed
Yeah.
data or patient level data or anything.
Doesn't surprise me, actually. Partly because of I think the people that they recruited, but also partly because of the endpoint, which is a very complex endpoint, but most importantly, the short duration of the study. You know, I think if you're going to use something like a chaperone for GBA1 activity, you have to do a trial over at least 2 years to see anything. So I think I would put that in the category of yet another poorly designed study that didn't show its endpoint.
Nick?
So, thanks, Tony. The microbiome could be a biomarker, but the other possibility, of course, is it's part of the process.
Of course.
So, and unlike the guy who gave himself peptic ulcers, you're not likely to want to give people the microbiome to get themselves Parkinson's, but you could do the opposite. You could treat the microbiome and see if you can drive these prodromals either backwards or delay their onset and that sort of thing. Has that been considered?
Yeah, absolutely. So what I didn't mention was that the signature in Parkinson's disease and in the prodrome is very significantly associated with the particular type of diet the individual has. So the good diet, you know, e.g. Mediterranean type diet, fruit, vegetables, etc., is associated with a non-Parkinson signature. A poor diet, high in saturated fatty acids, etc., low in fiber, associated with the Parkinson signature and, importantly, with progression, faster progression. So it's clear that the microbiome architecture is important in driving the nature of the disease, and there are many theoretical reasons why that may be. But yes, obviously, I think what a lot of people would like to do is see, can you change the microbiome and change the shift in the progression of the disease?
Okay, anything online? Great, thank you so much, Tony. So I'm really delighted now to introduce Miratul Muqit, who unfortunately couldn't be with us here in person, but is online. So Miratul, many of you may know, he's a professor of experimental neurology at the MRC Protein Phosphorylation and Ubiquitination Unit in Dundee. But Miratul is also one of the Aligning Science Across Parkinson's investigators, as well as a SPARC-NS investigator, a fellow of the Academy of Medical Sciences, and most recently has been appointed as the inaugural director of the UK DRI Parkinson's Research Centre. So Miratul really goes from basic discovery through to clinical application, and we're really pleased that he has accepted our invitation to give this plenary talk at the end of our first session. So thank you very much, Miratul. It's good to see you.
Thank you very much, Sonia, and I hope you can see my screen. And greetings from the north. And I'm really sorry that I haven't been able to join you in person. But thank you. I'd like to thank Sonia and the committee for asking me to give this talk. And what I thought I would do first, so I'd like to give a scientific update on my work. But what I thought would also be useful, I think, for this audience would be an exciting update in terms of a new centre for Parkinson's disease, which I was asked to lead last year. So what is the centre? So here are my conflicts. So what is the centre? So this is a National Centre for Parkinson's Research that's being jointly funded by the UK DRI, which is the UK's MRC-funded Brain Institute, and Parkinson's UK. And for me, the centre will be one that will be fully integrated with people with Parkinson's. So what is it that the centre wishes to do? We want to do breakthrough science, and we're really delighted that for the centre, it's really vital that we have the best people. And I think that the major highlight so far has been the appointment of these three fantastic scientists to the centre. I think the Henry Holden will need no introduction to this audience. Henry is a renowned gene hunter in neurodegeneration and neurological diseases, and his program will be to investigate the genetics, particularly of diverse populations in Parkinson's, and he will do that from UCL. We also have appointed Pete McGill, and Pete is the current director of an MRC core program in Oxford, but he's internationally recognised for his work in uncovering the circuitries that underlie basal ganglia homeostasis, particularly using mouse models to do that. And he will be trying to bridge molecular understanding of Parkinson's with systems behavioural dynamics in vivo. And then finally, I think many will also be familiar with Lara Parkinen, who is a translational neuropathologist that really seeks to take mechanistic insights and see if that can really lead to better understanding in human brains itself. So how will we achieve these breakthroughs? So we do need group leaders, we need individuals, but we wish to do this also through collaboration. And I think one of the major advantages of being embedded within the UK DRI is that we have access to nearly 30 groups across the 7 universities who are researching some aspects of Parkinson's. I don't have space in this slide to highlight all 30, but just to flag some examples of the depth of expertise in the UK DRI, we have, for example, Sarah Marzi, who's based at King's, who works on epigenetics in brain to understand both initiation and progression of Parkinson's. Recently has been awarded an ASAP program grant to understand the role of environmental toxins in Parkinson's. And then these two, I guess, will not be unfamiliar to this audience, Tim Bartels and Sonia Hong, who recently had a very seminal paper on the role of the gut-brain axis in Parkinson's, and Tim has internationally recognised research on synuclein misfolding in the disease. And then the UK DRI also has infrastructure for translation, including based here at UCL, the biomarker factory, which is led by Henrik Zetterberg and Amanda Hesselgrave. And then another major advantage is that we'll also be embedded in the ecosystem of Parkinson's UK, interacting with their own senior fellows, such as recent alumni of this includes Heather Mortiboys, who's now professor at Sheffield, Alistair Noyce, who's just down the road at East London. We also will have access to the Imperial Brain Bank, which is led by Steve Gentleman, and then the newly named Parkinson's Research Ventures, which is led by Lindsay Bilsland. And this for us is very important to take breakthrough science and accelerate the development of therapeutics based on that understanding. And finally, we don't wish to be inwardly looking. We want to be outwardly looking, and there's some fantastic initiatives happening all across the UK and globally in better understanding Parkinson's and leveraging that understanding for patients. And we wish to be also looking to build collaborative links with these initiatives. And I've just highlighted a few here, for example, EJS ACT-PD, and also there are several now other ASAP groups funded throughout the UK. And we'll also seek to put people at the heart of the centre, and we're finalising the recruitment of a People's Council. So this will be advertised shortly, where we'll look for people with the lived condition to contribute to what we do. So that's really a sort of a summary of what the centre is about, and happy to take questions at the end of this talk. I'm now going to move on to the science, and so my own lab has been very interested in the contribution of mitochondrial dysfunction to Parkinson's disease, and it's clear from many different types of experiments that mitochondrial dysfunction and the accumulation of this dysfunction is somehow linked to both initiation and progression of Parkinson's disease. And I think for us, I think that our work has really been underpinned by genetic discoveries in Parkinson's disease. I think for me, the two discoveries that have been most influential was it's been nearly 30 years since Nobutaka Hattori and colleagues discovered in Japan the Parkin gene mutations in people with early onset Parkinson's disease. This is just a figure from their paper, but one thing that will be relevant to my talk was that they highlighted in this paper that the N-terminal region of this newly found gene, Parkin, had very high homology to ubiquitin. So ubiquitin is a 76 amino acid protein that at that time was extremely well recognized for its role in protein degradation. So this was suggestive that somehow Parkin is involved in that process. And I've just highlighted here the residues which are conserved between Parkin and ubiquitin, including the serine residue, which I'm going to discuss later. And then more locally, the other discovery that's really benefited our work came from the lab of Nick Wood here at UCL, where he found mutations in this previously unstudied protein kinase called PINK1. It's an unusual kinase because it has at its N-terminus a signal peptide that localizes it to the mitochondria, and this was shown here in this figure. And then it also has a kinase domain, and you can see here with this annotation that the majority of mutations that cause Parkinson's lie within the kinase domain. And so this was something that really interested me at the time when I was in Nick's lab. I've left his lab and finished my training, and I decided to study this problem further. So before I actually started my independent research, there were several clearly established facts about PINK and Parkin, which I just want to summarize here. And I think these have largely remained true. So PINK and Parkin patients have very similar clinical features, including early onset of disease, high responsiveness to L-DOPA, and relatively slow progression, including, I think, a paucity of, for example, cognitive decline, which contrasts with the sporadic form. It was also it's also emerged that in terms of post-mortem analysis of people with PINK and Parkin, there are cases that do not exhibit Lewy body pathology. And then prior to my own work, it was demonstrated using Drosophila models of Parkin and PINK that Parkin and null flies have a very strong mitochondrial phenotype. And this was completely unexpected because Parkin is a cytosolic localized protein. And at that time, there were no clues that Parkin would have a mitochondrial role until this work in Drosophila. And then further work established that this was similar to the PINK1 null fly phenotype. And it was then shown that these two operate in Drosophila epistatically, in which PINK1 was thought to lie upstream of Parkin, but the mechanism of this was not really understood. So in terms of my own lab, when I started my lab, I moved to the MRC phosphorylation unit in Dundee, and I wanted to work on this protein kinase PINK1, and I was told by my mentors and directors at that time, Philip Cohen and Dario Lessi, the most important thing about a kinase is the identification of its substrate. If you find out what it phosphorylates, this will inform you on its potential physiological function. So that is what I sought to do, and I was quite focused on this. I remember calling Nick a couple of years into this project where we hadn't really made a lot of inroads, but I decided just to stick to this one question and not divert my attention to other areas. But it took us about 4 years to discover that PINK1 actually phosphorylates Parkin itself. Now you could argue that would have been predictable based on what I told you in the previous slide, but it turned out harder to prove that. It took us some sort of creative biochemistry to do this, but nevertheless, what we discovered was that PINK1 as a kinase, the reason why it was hard to study was it wasn't active under normal conditions, and so you had to activate the enzyme, and this we had to do using stresses that can damage the mitochondria. And we found that upon activation, it phosphorylates parkin at that N-terminal region that Hattori and colleagues had highlighted as being similar to ubiquitin. And we showed that it was phosphorylating this region at a residue serine 65. And we could show this here, for example, on this Western blot. Only when you activate cells with stress did you get the phosphorylation of parkin, and this isn't seen if you, for example, change the residue to an alternate amino acid such as alanine. So that was sort of the initial work that we contributed to, and then we made a very unexpected discovery a couple of years later, where we found that in addition to this residue on the ubiquitin-like domain of parkin, PINK1 can actually phosphorylate ubiquitin itself. And we found this, but also the laboratory of Richard Yule and Noriyuki Matsuda also found this. This for the ubiquitin field was a very novel idea. So ubiquitin was thought to be a modifier of proteins to target them for destruction, but it wasn't at that time thought that the modifier itself could be modified. But we showed that upon phosphorylation, this actually changes the function of ubiquitin, and you can see that here. So what we've done here in this experiment is that we produced the phosphorylated form of ubiquitin, and we incubated this in a test tube with parkin. And you can see here that when you do that, you get activation of the parkin enzyme, and this isn't true if you use normal ubiquitin. So that sort of proved the role of phosphorylated ubiquitin in activation of parkin. So these are biochemical experiments. So what does that really mean in terms of the cell and mechanisms of Parkinson's? And this is work that really came from other laboratories, particularly the laboratory of Richard Yule. And what he discovered, and this was confirmed by others, was that PINK and parkin are operating to sense mitochondrial damage and to then lead to the elimination of those damaged mitochondria by autophagy. So here is a cell, this could be a neuron, this could be a dopamine neuron, and in the course of the life of this dopamine neuron, there will be damage to mitochondria. And upon this damage, what Yule and others showed was that PINK and parkin cooperate together to remove that damaged mitochondrium. And this keeps the neuron or the cell healthy. But if you have a mutation in PINK1 or if you have a mutation in parkin, this damaged mitochondria cannot be removed. And so this leads to the accumulation of damaged mitochondria, which could then lead to further damage to the neuron in general and contribute to neurodegeneration. So this is a very simplified model, but I think it's actually emerged as one of the leading ideas for how Parkinson's can be initiated and progressed. And many other labs have contributed to this now, and this is sort of summarized in this figure. But I think the main players from this figure are to appreciate is that PINK1 itself is sensing the damage on the surface of mitochondria. Parkin, which is normally in the cytosol, is recruited by the activation of PINK to the surface of the mitochondria. You get the accumulation of ubiquitin, which is then phosphorylated by PINK, which acts as almost as the signal to the neuron that this is a damaged mitochondrium. And then this brings to that surface the autophagic machinery that then leads to the removal of this damaged mitochondrium. So that's great science, but what does that mean in terms of patient translation and therapeutics? Well, it's taken a while, but in this review that we wrote last year, we did flag that based on this fundamental knowledge, there are now some approaches which have entered clinical trials to boost the ability of PINK1 to remove damaged mitochondria, potentially as a treatment for Parkinson's. In one trial, which is being in phase 1 by AbbVie, this uses a small molecule which is thought to activate PINK1 directly in damaged mitochondria. That is in phase 1. And in a second trial, which has now entered phase 1b, there's another enzyme which I haven't mentioned called USP30. This is a deubiquitinase, so this acts to cleave ubiquitin chains. And so there are now one USP30 inhibitor for Mission Therapeutics, which is in phase 1b for the treatment of Parkinson's. And I think there will be others to follow. So one of the conundrums, however, remains this, is that despite very detailed molecular understanding of the activation of PINK1 and Parkin, there remains a very diverse age at onset of people that have either complete loss of function mutations of PINK1 or Parkin. In fact, this can lead to, for example, onset in childhood or onset as late as the 7th decade. And this cannot be explained by our current understanding of the model of PINK1 Parkin mitophagy. And this suggests that there must be other signaling pathways or other networks that are interplaying with mitophagy. I think there are some parallels here with, for example, the Huntington's field, where those that are clinically active will appreciate that people with Huntington's could be presenting at different ages, and this was thought to be regulate something beyond just the trinucleotide repeat length, and it led to the genetic discovery of the DNA damage components such as FAN1. I've talked to geneticists that work on PINK and Parkin, and they tell me that there are just too few cases of PINK1 and Parkin to do similar modifier genetic types of analysis at the moment. So I think we're very much left now to sort of perhaps produce candidate pathways by biochemical approaches. So in our work, what we decided to do was an siRNA screen looking for kinase modifiers of PINK1. And this was work that was led by 3 very talented postdocs at the time in the lab, Pawan Singh, Shalini Agarwal, and Ilaria Volpe. So what we did was very much a brute force biochemical experiment where we took HeLa cells, which we damaged with mitochondrial stress in the presence of siRNAs for nearly all known serine threonine kinases. And to cut a long story short, this is basically an example of the screen. It was a Western blot-based screen, but what this produced was a very clear result. And what we found was that there was one kinase called EIF2AK1, also known as HRI, which if you knock this down, this led to an increase in both PINK1 levels and PINK1 substrate phosphorylation, which you can see, for example, here with the phosphorylated ubiquitin. So what is this EIF2AK1? Well, it's a member of a stress response pathway that's very well studied in biology, also known as the ISR pathway. There are 4 member kinases of this pathway. Probably the most famous is the ER stress kinase PERK, so this responds to ER stress. There's also PKR, which responds to viral DNA. And then there's GCN2, which responds to changes in amino acids in the cell. And HRI was previously thought to be purely sensing changes in heme or iron, but recent work suggested actually it had other functions. All 4 of these kinases phosphorylate this translation factor called EIF2 alpha, and upon its phosphorylation, this leads to a general reduction in translation and an increase in this transcription factor ATF4. And what this does is this enables the cell to essentially conserve energy for combating stress. So what we showed initially was that the effect on PINK1 was purely driven by EIF2AK1. So if you knock down the other members 2, 3, or 4, we do not see these effects on PINK1. And in terms of what the mechanism of that is, this was work that was done by the Campman and J labs. They discovered a very novel relay that operates between mitochondria and cytosol, and it's mediated by this previously unstudied protein called DELI1. So DELI1 normally sits in the mitochondria and is inert, but upon mitochondrial stress, it's cleaved by a protease called ERMA1. And the cleaved fragment of DELI1 is released into the cytosol where it activates HRI. And we can show in our study that if you knock down HRI or DELI1, you get the same effects on PINK1. So we think that the PINK1 upregulation is mediated by this DELI relay system. And we were then very fortunate to collaborate with Ian Ganley, who's a PI here in the MRC phosphorylation unit, and his very talented postdoc Leah, to show that not only does this change PINK1 signaling, but it also has an impact on mitophagy. And you can see that here. This is an assay that Ian Ganley's labs developed called the MitoQC. How it works is that when you have mitophagy, you get the accumulation of red puncta. And what Ian was able to show here was that you got increased red puncta when you knocked down the EIF2K1 HRI kinase, and this was quantified here. So what this work suggests is that simultaneously in damaged mitochondria, not only do you get activation of PINK1, but you're also getting activation of other stress pathways such as ISR. And this is acting as a negative brake to mitophagy. It may be that in some situations, the neuron doesn't want to get rid of the entire mitochondrion. Maybe this is a way for the mitochondrion to maybe initiate other local repair rather than complete removal. But this is work that needs to be further extended and worked out. But what I think is interesting from a therapeutic point of view is that the ISR pathway, which I mentioned is extremely well studied in biology, has in its own field come up with several drugs that can modulate it. And one of the very first drugs, this was developed by Peter Walter's lab, who's one of the pioneers, is called ISRIB. So ISRIB inhibits the ISR. So we took ISRIB and looked to see what happens if you inhibit this ISR inhibition. And what we saw was that, as expected, using ISRIB led to an increase in PINK1 signaling, and therefore, and we also then showed mitophagy. So we're very excited that this new understanding could actually have some immediate clinical translational implications. And in particular, there are now better analogues of ISRIB. In fact, there was a recent clinical trial in ALS that was negative, which used two ISRIB analogues, one that was from AbbVie and Calico and one that was from Denali. And I'm assuming that these are both CNS penetrant. And we currently are testing these compounds in our laboratory. And if these do show effects on PINK1 mitophagy, then I do think these would be very interesting molecules to test in potential clinical experimental paradigms. So I'm just going to finish by thanking you for your attention. As Sonia mentioned, I'm funded by other organizations as well. And for the younger members of the audience, I just wanted to flag that we recently were able to obtain a ASAP award. And this is a multi-center team with colleagues in Germany, UK, and in the US, both basic scientists and also clinicians and clinical pathologists. And we're currently recruiting, and it's going to be on fundamental understanding of mitochondria in Parkinson's. And if there's anybody interested, then please get in touch. But with that, I'd like to finish, and I thank you for your attention. Thank you very much.
Huge thanks, Miratul. And thank you also for giving us the overview of the Parkinson's Research Centre, as well as that great work. Any questions for Miratul in the room or online? So Miratul, tell us where, the integrated stress response, as, the mitophagy was sort of almost the way into that. do you think that's going to be something that can be modulated? It's such a broad sort of host response, really, to whether it's organelle damage or whether it's proteostatic stress. So I'm just wondering how easy you think it will be to use that as a sort of modifiable target.
Yeah, so I think that the ISR response operates in every cell and is very diversely regulated. So having a broad ISR inhibitor, I think, is not going to be useful. I think, for example, in Parkinson's, if we were to assume that mitophagy operated or is a key mechanism in sporadic Parkinson's, then I think you'd have to focus on the specific regulatory elements that we, for example, have identified, like DELI1. So DELI1, we think, only seems to be working when there's mitochondrial damage. So I think one could, and we're also working to try and elaborate even further the other players in the mitochondrial ISR. So I think, Sonia, if we were able to get more of a better understanding of the molecular sort of machineries, if you target those, I think those could be more specific for Parkinson's as opposed to, say, other diseases.
Yeah. And what do you think about the relevance of those processes in sporadic disease? I mean, obviously we've got to them via the PINK1 and Parkin forms of the disease, but so much of the treatment is really for the sporadic Parkinson's population.
Yeah, no, I have to say, I mean, that's still an open question, which is PINK-Parkin relevant to sporadic or idiopathic Parkinson's disease? I think our view of Parkinson's is a very alpha-synuclein, of sporadic at least, is a very alpha-synuclein-centric view. But we have we're just working on a new study which we hope to hopefully drop in the next few months, where we've easily confirmed the work of Virginia Lee's. So Virginia Lee in 2003 showed that in Parkinson's disease, the levels of Parkin are significantly reduced, particularly soluble Parkin, and there's an accumulation of insoluble Parkin in the brain. This is also work that Michael Schlossmacher has shown, and we've actually been using brains. This is in collaboration with Tom Warner and Zani, so we've used brains from Queen Square. We can easily replicate that. So the decrease in soluble Parkin in sporadic Parkinson's is real. So I think that would indicate that there is possibly some contribution of this to the disease.
Great. Okay. With that, I'm going to say a huge thank you, Miratul. Thanks for joining us online, and give you one more round of applause.
Thank you.
Okay, and with that, we can close the first session. What a great start to the day.
I'm Hugh Morris. I work here at UCL in the Movement Disorders Centre. It's great to see so many people here. It's great to see so many young, enthusiastic people here, and also old, enthusiastic people as well. So a spread of all generations. We've heard some great talks, and obviously we've been thinking in part about Parkinson's disease, which of course is extremely important in terms of number of people affected and what we want to do to try and change the natural history of Parkinson's. But also rarer movement disorders are important as well, both in terms of how they affect people, but also in terms of what we can understand about basal ganglia biology and function based on the physiology and the molecular genetics of these conditions. Great pleasure to welcome my friend and colleague Professor Kailash Bhatia as the next speaker, well known probably to everyone in the audience. Leadership in the Movement Disorders Society, leadership in rare disease, rare movement disorders research, both in genetics, clinical phenomenology, physiology, president-elect, I think, of the EAN still currently. So it's a great pleasure to welcome him to talk about rare movement disorders, unmet needs and opportunities.
Thanks so much, Hugh, and Sonia, thanks for this invitation. It's really great to see this audience today. So many also current but also past fellows and colleagues and friends here today. So as Hugh was mentioning, one of my interests for a very long period of time has been in rare movement disorders. But I think we probably need to sort of start with the question, do rare diseases matter? I remember in the past, when with this interest, Peter Goadsby telling me, you're just a stamp collector. So you have a little album and you put new stamps into that. But if you think about rare diseases, and you heard about Gaucher's this morning, a rare disease which has had profound pathophysiological and other implications in the day-to-day Parkinson's disease. So in Europe, rare disease is defined as fewer than 1 per 2,000 individuals. And there are over 6,000 rare diseases. An important thing to note is 70% or so are genetic. And a lot of them are of pediatric onset and so on. And if you sort of think about it as each rare disease having a very low prevalence, or really does it matter or doesn't matter, but if you put this together cumulatively, and then you think about that, then about 6% population globally is affected by rare disease. So we're talking about something like 450 million people who are affected. And I remember when I was a fellow having a patient who I very sort of, enthusiastically had diagnosed as having a brain iron accumulation disorder. And then she asked me, the mother, so what's going to happen to my daughter? Why is there iron in her brain? Is she going to progress? Have you got a treatment for her? And I had none of those answers. And her question to me was, why isn't there any research into this? And the answer was that there are very few people affected. Maybe the drug companies are not interested. Maybe you can't get funding for these sort of things. And I think those questions still apply. And I keep that in mind when I'm seeing this. So the other thing is about equity. And the more I'm sort of getting into the EAN and so on, I'm thinking more broadly nowadays about public health and equity, and actually the lack of equity with regard. So we live in these ivory towers, but there's a big world out there, and there is inequity. So this is a problem not just affecting Europe. We got about 30 million people here, but this rare disease affects all over the world, and there are millions and millions of people who are asking these questions, which that lady asked me the very first time I diagnosed it. And this is from last year. The World Health Assembly and the World Health WHO has now adopted a resolution. So you may not know about this, and this makes rare disease a global health priority. So this is a strategic framework because it's, as I told you, more than 300 million people are affected. So there's a 10-year action plan suggested by the WHO. National policy integration, so member states are asked to integrate their rare disease policies into a national health system. So this is now, and the UK is a signatory of this, and one of the important things, as I said, is equitable care with regard access to affordable medicines, technologies, and diagnosis, and where to go if you have a disorder like that. So, as I mentioned to you, and we don't recognize this, we don't realize this, there are disparities in the diagnosis and management of rare movement disorders. So I'm talking about movement disorders. And if you look over here, access to genetic tests in movement disorders, Parkinsonism. So really, in Europe is not bad, 50%, but Africa, almost negligible. And each of these things in all these different disorders, so yes is available, which is dark blue, no, not available, and light blue, unknown. So you can see that there are difficulties with regard to genetic testing almost all over the world, but particularly in Africa and Asia. Then you have also these challenges about access to general neurologists, access to a movement disorder neurologist. Many of these disorders are pediatric in origin, access to pediatric neurologists. And there's a lot of problem here, even in Europe. So if you're thinking about Europe, you say, oh yeah, well, we are doing well, but actually we are not. There's a lot of disparity here. In Queen Square, if I'm ordering a genetic test, it takes me a year and a half sometimes to get the result. So imagine telling your patient, I think you've got a genetic disease, it's a rare disease, I'm going to send you a sample, but you'll get a result after a year and a half. That's not acceptable. Imagine if you were the patient and you're thinking about this for the whole year, have I got it, have I not got it? I'm waiting for a year to get an answer. The other problem is that we have getting geneticists, or you have a genetic diagnosis, genetic counselors. You need genetic counseling to be able to give the diagnosis to these patients. And that also is quite difficult and not available in many, many countries. And nowadays, with the NGS and so on, we have the diagnosis available, maybe possible, but who's going to make that diagnosis? Who's going to do the genetic counseling, which is extremely important in these conditions. So that's one about diagnosis. But even management of rare movement disorder diseases in different world regions is a problem. And if you think about it, genetic testing, and this is Africa here, and these are many of the things we take here for granted. MRI scan is available, that scan is available, FDG PET scans are available, but this is not readily so in many parts of the world. So genetic testing in Africa, for example, difficult, no availability of many of these conditions. So the DAT scan, which you do all the time, not available. Is that really important? Are you going to make a clinical diagnosis? In some situations, it is important. And the same thing also applies in Asia, Oceania, and Pan America, that we have difficulties in this, getting many of these tests which we take for granted available. So this is sort of a little summary of this unawareness of these disorders. Many of the GPs, many of the local neurologists or physicians are unaware of these conditions. And that's why you need access to specialist centers. But where do I send it is another question. Okay? Scarcity of movement disorder specialists. Concentration of movement disorder specialists in the main cities. So over here, we have many movement disorder specialists in Queen Square. But in many of the other regional centers, there may not be many of them. Limited access to specialized centers. And limited access to diagnosis and therapeutic tools, even if you arrive at a specialist center. So this is a problem which we need to be thinking about. And that was for the other countries. The same thing also applies over here in Europe. So this is work done by the MDS, Marina Tissin's group, telling us that there are problems also in genetic testing, not readily available, management of patients, accessibility to tertiary hospitals, to experts, to multidisciplinary teams is unequal between countries. Germany is doing very well. Some countries are doing very well, but it's not available everywhere. Even things like botulinum toxin, we heard about advanced DBS and so on, but these are very few and far between. And these are treatable disorders. So you saw that you can get really good results in people with dystonia, for example, with DBS, but good DBS, we have a problem. So the management of these conditions, even in the EU countries, is unequal. And then you sort of ask yourself another question. Is there clinical utility of all these advanced genetic testing or so on? If it was available to you, does it make a difference? That's another question you need to ask. And here is the yield, so a diagnostic yield of genetic testing in movement disorders. And these are a mixed group. So this is a mixed bunch of people here with different movement disorders. 21%, 32% is the yield. You may say that's small. But in those 20 or 30%, you made the diagnosis. And there may be treatment implications, which I'll show you in just a minute. The other interesting thing is that we get a lot of variants of unknown significance or genes of unknown significance. And interpreting that is also a problem for the genetic field. And that's something we have to also keep in mind. How do we improve that on that? See, in the ataxias, for example, the yield seems to be much better than in dystonia. So particularly for the classic ataxias, 50% is not a bad result. And if you're thinking about Parkinson's disease, it's low, the diagnostic yield, about 9 to 20% in different centers. But a number of variants of unknown significance, up to 22% in some of the studies. So this needs to be looked into as well, maybe an international consortium. And there is something like that, but looking at these variants of unknown significance, maybe having more advanced ways of looking at it RNA sequencing, etc. There are many other tools, but just an interesting observation. And now that you have, I told you, does it make a difference in those 15 or 20 or 30%? The answer is yes. And there are now over 70 disorders which are genetic conditions in movement disorders where you can make a contribution with regard to their therapeutic or treatment implication. So you may have targeted medication. I'm just going to go through all of these, but if you think about aceruloplasminemia, iron chelation, fresh frozen plasma, dopa-responsive dystonia is obvious, benign heterochorea, levodopa is helpful in benign heterochorea. Okay. Friedreich's ataxia, we've all heard about this. Hypermagnesemia, manganese chelation with EDTA, and I've had patients with that, and they have completely improved, like completely gone. Their dystonia, Parkinsonism, manganese in the brain, manganese chelation with EDTA, completely recovered. So gratifying. So but you have to make the diagnosis. Niemann-Pick C, miglustat, and now N-acetylcysteine, and so on. Rapid onset dystonia, Parkinsonism, flunarizing. So, like I said, I'm not going to go through all of them, but there can be implications directly with targeted medications or dietary modification. So you may change the diet. Ataxia with coenzyme Q, you may give coenzyme Q supplementation. Biotin, thiamine, responsive basal ganglia disease, biotin and thiamine supplementation. Folate deficiency, glutaric aciduria, and all these have important this. Dietary modifications. I remember my patient with GLUT1 deficiency, with ataxia, with paroxysmal exercise-induced dyskinesia, and giving them a ketogenic diet completely abolished the exercise-induced dystonia, which was a real problem for that young lady. Gene therapy, many things coming out, but the one which we are now talking about is AADC, fantastic results in that, these children with oculogeric crisis, etc. Trigger avoidance. So you made the diagnosis with many triggers which you can avoid. So there are therapeutic implications in making the diagnosis. We heard about DBS just now. And nowadays I would say to you that if you're considering DBS for dystonia, you must know the genetic makeup of your patient. Because certain genes do well and other genes don't do well. Some do even worse, that they go even, so the DCAF17, they get really very bad. And you can see there's a lot of difference. So DYT1, amazing improvement, but that may not be the same for others. So DYT3, not really good. ADCVI5, not very good. ANO3, excellent. So your genetic makeup, is very crucial with regard to the outcome also for DBS. And this may also apply to many of the Parkinsonian conditions and other disorders. So I also thought that this gives me an opportunity to really sort of showpiece at least some of the important genetic discoveries we made here in our group. And I've really been, I've really had great collaboration with my colleagues here in neurogenetic. They've always been friendly, always been cooperative. Nick Wood, a friend, really good colleague, John Hardy, Henry Holden. And I just thought I'd just tell you what we've been doing so far. Many of these conditions which we have described from here, patients from here, so mutations in NO3, and also looked at the pathophysiological mechanisms, the protein structure. And we're also talking about possible disease modification, etc., to come in the future. So these two important ones, Gavin Charlesworth here, who I worked with Nick Wood and myself, found two important genes here. This was a family which had been seen even by Anita Harding's time, no diagnosis made, and we were able to find that they have the HPCA, autosomal recessive, primary isolated dystonia. ANO3 is now one of the important causes of crâniocervical tremulous dystonia, which Francesca Morgante was showing you. Some of them may have ANO3, and as I said to you, the ones who do well, ANO3 does well. Phosphodiesterase E10A, this is Niccolo Mencaci. Again, a number of these patients with striatal necrosis and what looked like benign heterochorea.
Would you be able?
And ADCY5, also by Niccolo Mencaci. And some other rarer dystonia genes described very good publications from our group. And most recently, Francesca Megrinelli here has found the PSMF1 as a cause of Parkinsonism, and we have a very large number of cases in this paper showing that these biallelic missense mutations, and you can have this phenotype with early onset PD Parkinson. This has just come out, and on one side, and arthrogryposis on the other side, and in between stages. So really fantastic. She's done an excellent job about the protein structure, the mitochondrial implications, etc. You can look this up. All of these things require partnerships and networking too. So we are members of the European Reference Network as a collaboration rather than a proper member. Because of Brexit, we can't be a proper member of the ERN. ERN is really dynamic. And one aspect of ERN is the Solve-RD. So this is a fantastic enterprise where they are putting together, trying to solve rare disease, which nobody else has been able to solve. And Francesca Megrinelli has recently found the gene for hereditary geniospasm. She's not revealing it yet, but we shall hear about this very soon, and this was done with the Solve-RD people. We also have international partnerships, so defining better the nomenclature, the diagnosis, evaluation and management, because these conditions are rare. So you need to put together consortiums so that you can have larger numbers, so you can understand better the nomenclature diagnosis. And we've been doing that for primary brain calcification. I just published this recently. And there's a UK a charity called Far Beyond for brain calcification. This is Adam Tate, who was one of my patients. Orthostatic tremor group, and Amit Batla is the president of this. So we're doing some work in orthostatic tremor as well. And the NHS England, and I'm really very pleased about this. So we have set up the Queen Square Rare Movement Disorder Collaborative Network, and Sonia Gandhi and Chris Lambert, Amit Batla, Pras, Francesca and Manju Kurian, we are members of this. We have rare movement disorder, genetic and other disorders, MDT meetings. So if you want to submit a case, we are very happy to look into that. And this collaborative network, we are hoping that this would be a pathway for people to refer cases to us from the whole of the UK, and we can try and solve them. So we want to provide expert diagnostic service for local and national referrals, provide advanced genetic testing and counseling, other investigations that might be needed, telemedicine or remote advice, and research genetic testing in those who are undiagnosed. Maybe this will lead to better understanding of mechanisms and pathophysiology, and we are forming trial-ready cohorts of rare conditions. We are thinking about working with pharma and industry for clinical trials, and we are exploring philanthropy and crowdfunding for particular projects. So I had this patient with SCAR12, and SCAR12 is seen in the particular Indian community called the Agarwals. And when he came to us, he had a tremor which had been diagnosed as essential tremor, but because of his surname, which was Agarwal, and we tested him for SCAR12, he was positive, and he said, what's going to happen to me? He said, well, you're going to progress. Am I going to become Parkinsonian? Some people do become Parkinsonian. How are you going to stop this? So we can't. There are only about 100 cases around the world described in the literature. And he says, what can I do for it? Can I set up a crowdfunding for you to find a cure for SCAR12? He says, sure, yes. So, may have some targeted projects for things like that. And just talking about this, about the FAST study, so we have just started this, the calcifate trial. This is for patients who have calcification of the brain from irrespective of the cause. So you can have any of the genes which are causing basal ganglia, cerebellar calcification, and we are using etidronate, which is a biphosphonate, which slows the bone resorption, and it has been shown to be useful in other types of calcification. So this trial is on at the moment. And if you have a case you want to submit, you can you can send it to Francesca Migranelli here. The inclusion criteria about 18 years, clinical diagnosis of FAS, and bilateral classification of the basal ganglia. So screening visit, telephone call, blood tests, simple study, 1 year follow-up, and so far we have got 10 patients here. This is along with the University Medical College Utrecht, who are the sponsors of this study. So our target is about 30 patients. So if you got somebody with FAS, do reach out to us. So I again just want to come back to what I just started with. The consequences of these disparities, and can we do something about this, which I mentioned to you earlier. And as I said to you, there are a number of issues: delayed diagnosis, wrong diagnosis, multiple interactions with various specialists before you actually arrive to the right person and the right diagnosis. So these disparities are an issue which I think we really need to think about. They affect quality of life of these patients, negative balance on work-life balance, and we do have some treatments, but reimbursement for these orphan drugs can be quite expensive, and this varies across different countries. So the suggestion from this paper was that these are the recommendations which they had made: ensuring research and development of essential evidence, encouraging investment in research, building equitable access to diagnosis and treatment, building capacity. So we are hoping that with this, at least we made one start here in the UK for the Rare Movement Disorder Collaborative Network. And as I said to you, you can send us patients and we'll do our best to do something about them. Thank you very much for your attention.
Thanks, Kailash. Great talk and a great overview of the sort of global situation. It's a great pleasure to welcome the next speaker, Professor Lynn Rochester from Newcastle, as Professor of Human Movement Neuroscience. I've known Lynn for quite a few years, and going back a few years, Lynn was a huge kind of proponent of using digital measures to measure movement and to use that to monitor patients and to use that for outcome measures. I think it's fair to say that she was ahead of her time, and initially people didn't think this was a going concern, but we can see an explosion of digital measures and incorporation into clinical trial designs, and really look forward to hearing from Lynn about updates in this area.
Thank you for that kind introduction, and thank you very much for the invitation to talk to you today. I'm really delighted to be here. So let me just orientate myself. So I'm going to talk about developing the next generation digital measures for trials and care. I'm focusing on the work of the Mobilise Consortium. So there's a clue in that. I'm going to be focused on mobility measures. These are discrete measures of gait and activity, and particularly how we measure in the real world and measure continuously and passively. And this really chimes with the last two speakers, this need to have an inclusive and equitable access to robust tools to help us evaluate the work that we do clinically, but also our research work. So to begin with, why gait disturbance? Why is this an important area, and why should we be monitoring it? Well, we know this is work from our group in Newcastle, that gait is, we see gait impairments at diagnosis, and we see very specific gait impairments progressing even in spite of optimal medication. In fact, we see impairments in gait in the prodromal period, which progress up to the point of diagnosis as well, from work in collaboration with Walter Maetzler. So these are measures that could potentially be telling us something important to help in the detection and monitoring of Parkinson's. We also know, as mobility loss is a core feature of Parkinson's, that and a highly problematic feature of Parkinson's for patients, that this is something where they want to have focused research energy in order to make this a priority to have treatments to mitigate mobility loss. So for these two reasons, better measures in the management of Parkinson's and also measuring something accurately that patients care about, it's really important that we're doing this and doing it better than we do now. Another really important feature, if we think about clinical trials, is that there's a widely acknowledged limitation in the scales and the approach that we use to measurement in clinical trials. And this is really nicely summarised by Sonia Mather, who's a GP living with Parkinson's disease in Canada, who highlights the problems of a mere clinical snapshot, which really doesn't do the job when you have a highly fluctuating and heterogeneous condition such as Parkinson's. And also highlights the opportunity and the benefits of more long-term monitoring that arguably could capture the lived experience with more relevance than the tools that we use at the moment. So if you think about the ability to monitor mobility, gait and activity in the real world, this and to do this passively, arguably this would bring a highly appropriate measure of what matters, but also do this where it matters, delivering more relevant, personalised and sensitive tools to help us manage and drive research forward, innovate. So this is comes to the case for mobility or digital mobility outcome measures as measures. And we know that wearable devices can quantify digital mobility outcome measures. I'm sure you're all carrying them in your pocket or wearing them on your wrist. With improvements now in battery life and sensors within these devices, this has really driven the ability for long-term passive monitoring. We've heard about access. This also provides the opportunity for inclusive approaches because it's not limited by geography. We do work in sub-Saharan Africa and other areas. So this really gives us an opportunity to be more inclusive in who we can measure, who we can monitor and where we can do it, and really chimes with the NHS 10-year plan as well. And these measures are objective and more precise, arguably, than the tools that we use. And of course, this, as I've just said, brings the opportunity to really drive innovation in clinical trials, but arguably to drive innovation in clinical decision-making and how we manage our patients, addressing the problems that they really care about rather than more abstract symptomatic aspects. However, despite what you may think about your various devices, I won't name any, prior to widespread use, we have to validate these tools, particularly if you think about why we're using them. And the majority of devices that we have access to are simply inaccurate. And this was the motivation for the Mobilise Consortium, which was formed. This work started all the way back in 2017, in fact. And this was raised as a funding area by industry who really wanted to adopt the use of these tools in their clinical trials. And they wanted to develop a solution, digital mobility outcomes, as endpoints for use in clinical trials and care. This was funded through the EU, through the IMI initiative, which brings together academic partners and multi-stakeholders together with industry to address these big questions. And I would say that this type of work is really only possible with this nature of funding, because of the complexity that's required to deliver on these sort of things. So we had 34 partners in Mobilised. We had a very large budget, as you can see there, to deliver on what was a very ambitious agenda over a 5-year period. The industry partners, thankfully, also added an additional 2 years of funding to allow us to continue interrogating the gigantic data set that we've generated within Mobilised. It's a highly multidisciplinary consortium of the scale needed for this type of work, so multidisciplinary academics, healthcare professionals. We had patient representatives involved all the way through, and obviously this was a private academic interaction as well. All of this necessary. So the aims, I've really just said this, we needed to develop a digital technology solution to continuously assess real-world mobility. Imagine trying to measure your gait speed as you go about your day-to-day activity or your stride length or any other discrete aspect. So this was a challenge. This is a real-world challenge. We had to do this using a single body-worn device. The algorithm that we developed had to be able to work on different devices, so we didn't get to the problem of the device company failing and then the entirety of the research is lost. And we had to demonstrate that this was an effective way to measure mobility across a range of different conditions. And you can see our conditions here. And these were all chosen to really test the algorithm because mobility is mobility. We don't need a different mobility scale depending on your condition. We should be measuring, using the same tools to measure the same thing, but have the devices that can work in an accurate way. So obviously providing a generic and harmonized approach to mobility assessment. So we had a very ambitious objective, which was a little bit naive thinking back, which was to seek regulatory qualification of digital mobility measures as secondary endpoints in clinical trials and care for use generically. And this really was essential that we kept sight of this, because the standard required, just as you develop a new therapy, the standards to approve that therapy are very high. It's the same for developing a new measure. It has to be of the right standard in order for this to become a generic tool. To be clear, or just to tell you what our mobility measurement framework was, we had a very functional definition of mobility. I've just said it before, that's walking as you carry out your day-to-day activities. And the converse of that, mobility disability, being a limitation in your ability to do that. And this was our meaningful aspect of health, which of course we need to demonstrate as a clinical outcome assessment measure to show that you're changing how a patient feels, functions and survives. We would measure this using our digital tool that we've developed as a measure of mobility performance, what someone is actually doing as they go about their day-to-day activities over 7 days with a wearable device. And you can see one of the devices that we used in the consortium on this being worn by this lady here. So mobility performance was our concept of interest in regulatory speak. We weren't looking to measure one thing. A device generates a raw signal from which you can measure many aspects of function. So we took a broad approach. We were measuring walking-related activity, and you can see some examples here, as well as very discrete gait analysis, and there are some other examples there. And these were our digital measures, or that eventually, hopefully, will be endpoints. And then this is the context of use that we had defined in Mobilised, but of course there are many other contexts of use to deploy these measures. But this was really focused on the evaluation of therapy that would slow the progression of mobility disability as a secondary efficacy endpoint to begin with. So Mobilised had two grand challenges that it had to meet. It firstly had to show, we had to develop an algorithm, because this hadn't been done, and we had to show that the algorithm paired with a device with the right technical characteristics would accurately measure mobility. We had to demonstrate this within a technical clinical study of 120 people across 6 cohorts, and we had to demonstrate and validate this in the real world, accounting for a range of confounding measures. And then we had to show in our second grand challenge that these digital mobility outcome measures across all of our cohorts told us something important about health status. They could discriminate by condition and by severity, they could monitor change over a period of time, and they could predict outcome. Importantly, voice of patients was included throughout all of our work in order to ensure everything that we were developing was meaningful, acceptable and informed. And we were speaking with the regulators from before the start of the Mobilised consortium work, because we had to ensure that all of the methods that we were innovating and developing were going to generate the level of evidence that would ultimately seek regulatory approval. And in Parkinson's, we were fortunate as well to gain additional funding to carry out an extension study that's been led by Alison Yarnall. I should have said I was the coordinator of Mobilised together with Novartis. I've just realised I've forgotten to say that. And this allowed us to collect data in a large control cohort to inform progression against normal ageing, as well as having an additional time point at 36 months in Parkinson's disease, and to allow us to inform future healthcare use. So very, very top level of what we found, because there have been literally hundreds of people involved in this work, so I can't reference papers and individuals, but just a high level. And before I do that, this is a quick exam question for you. I want you to look at this video and tell me how many steps Razor takes. that was the true step count. Who got that? Oh, well done. However, we monitored him with a range of different devices, and you can see how variable that step count is. Imagine if you're using these and interchanging your tools when you're evaluating patients. This is still a problem in technology, and you will still not see people validating their devices appropriately. Different wearables report different outcomes, and validation, robust validation, is absolutely essential. And that's what we did in the technical validation study. And cutting to the chase of 3 years of work from hundreds of very talented researchers internationally, we were able to show through the algorithm development and a study conducted in 120 people, a clinical technical validation study, that the device and algorithm pair that we had designed and developed would accurately quantify a range of different digital mobility outcome measures across all of our cohorts of interest to a level of accuracy that was acceptable. The methods were acceptable to patients, but also to the staff deploying them. And the tools that we developed were device agnostic, so we could run these on different devices, and we tested this as long as the device had the right technical characteristics. We also had a device company fail on us, so we had to do this by necessity rather than desire, as it turned out. It's really important in these large consortia to drive as much impact as possible because of the amount of funding, and we have had this in mind from the beginning. We've shared our data. It's available on Zenodo. We've shared our algorithm under a commercial and academic license. That was quite a brave move, but I'm really delighted to say that these methods now have been adopted by 4 different companies, are bringing these to more widespread use. We've also developed a tutorial, the technical implementation manual, and all of the engineering detail that went into this can be accessed through this technical implementation manual, and you can find this through our website as well. So moving to the clinical validation study, we obviously had all of our cohorts and enrolled 2,400 participants across 16 sites in Europe. To determine clinical validity, but in Parkinson's we had 5 sites, and you can see them represented on here. And for each of those sites, we collected data in 601 people with Parkinson's disease. They all underwent a digital mobility assessment and then had the appropriate clinical assessment and questionnaires at the same time. So they were assessed every 6 months over a 2-year period, and we've got an additional time point in this particular cohort at 36 months. We collected data in 220 age-matched controls as well at baseline and 12 months, so we've got this control reference point. And initially, in our first tranche of outcomes, have looked at 24 DMOs that are undergoing evaluation, but I can tell you that many more are being tested now as well. Importantly, we established reliability of those outcomes so that they're stable when they need to be, and we want to know that they change when they should, and they're not doing that if they don't. You can see that the PD cohort is diverse and representative, as we would expect. They're mainly on the mild side, with the majority of people being Herniar 2. We're seeing falls in the anticipated frequency that we would expect, and this cohort was well matched to our control cohort. So just a snapshot of what real-world mobility looks like in Parkinson's. We can see that people with Parkinson's, unsurprisingly, walk less. They take fewer steps. They walk for a shorter duration, and they walk with a slower speed compared to our control cohort. Those eagle-eyed of you in the audience might be looking at that step count and thinking, that's a pretty impressive step count. But that is because we're accurately measuring steps rather than pedometers and health-related thresholds of 10,000 steps a day that have been in the literature for many years. Those thresholds need to be reset because they are not based on accurate step counts. These measures are based on accurate step counts. So it's something for wider aging and health to consider. Just thinking a little bit more about clinical validity, I can only show a very small amount of data also, looking at the time. But one of the things that we wanted to do was to see if these were going to be useful to discriminate. All of our outcomes, we have our 24 outcomes here represented as normalized data. The control cohort are in black in the center. All of our outcomes discriminated Parkinson's from the control cohort when identified together. But more importantly, if we think about the ability to discriminate between controls and early Parkinson's disease, 21 of these 24 measures were able to significantly discriminate. And this is important when we think about the potential for neuroprotective trials in the future. If we think about the ability to discriminate between disease stages, such as early to more moderate disease, the Hernon-Yar 1 and 2 compared to 3, we also see significant differences in the amount, variability, pace and rhythm of walking as well. So 12 of these outcomes. And again, this is potentially important for disease-modifying therapies in the future. And there'll be many more findings to come in, over this. We are currently looking at our longitudinal data, so this is a snapshot of work by Alison Yarnall and Cameron Kirk, preliminary analysis, and I'm just showing one outcome here, changes in walking speed. And we can see that walking speed is indeed changing over the 3-year period and on a year-on-year basis. The important questions for us that we are currently answering is to look at the rate of change in this particular outcome measure compared to the rate of change in the UPDRS scales, because what we would like to do is to start to think about these as responsive measures and think about how they may underpin go, no-go decision-making within clinical trials. But this is the first step in the in monitoring and a movement towards responsiveness for that application. Very briefly, clinical validity, our approach, even though it was a body-worn device and not a wrist-worn device, and I know there's lots of debate about that, but compliance was high for devices, and you can see some data here. We had really excellent participant engagement. We had some loss of DMO data from the baseline testing, which we learned very quickly and prevented that from happening, so that the majority of our cohort had a full 7 days of data in their data monitoring. We're also sharing this data at the end of June for wider release, and that will continue to generate impact from Mobilise to allow the wider community to continue innovation with these digital measures. Now, I said that we were interacting with regulators at the beginning, and this was key to adoption. And we were fortunate that they approved in principle the approaches we were taking to technical and clinical validation through two letters of support. But they did highlight remaining evidence gaps if we want to go for qualification. And these were the question of meaningfulness for what patients call importance, the question of meaningfulness of mobility performance. If you want a new therapy, you have to show that it changes how someone feels, functions, and survives, and that's exactly what this is talking about. And from the work that we carried out, we can show that mobility not only affects people physically, but has profound mental and social effects, as well as sense of self and the ability to interact in the wider world. So it's very, very important. We had acquired some additional funding again from the MJ Fox Foundation, and that's allowed us to relate our mobility outcomes to symptomatic changes, but also our processes. And this has been confirmed in multiple underserved groups as well, showing that these generalize rather than are discrete to a very defined group of people. And then we're relating now, carrying out work to relate the DMOs to discrete symptoms as well. The other gap is responsiveness. You can only produce the evidence for responsiveness within a clinical trial. And I'm absolutely delighted to say that the digital Mobilise has been included as a digital measure sub-study in the EJS ACT consortium led by Camille Carroll and Tom Foltynie. This particular sub-study is being led by Alison Yarnall, and this will generate another body of evidence to inform go, no-go decisions and support the final step for regulatory qualification. And then my last slide, you can generate millions of outcomes from these devices, and you can have proxies of sleep and fatigue and all sorts of different things. So the data set will continue to generate innovations. We continue to do some work. This is work from Lisa Alcock and Claire Wilkinson. That's validating a turning algorithm, which is a very sensitive metric. And we're continuing the evolution of digital measures as well. So this space is only going to increase in activity. So in summary, mobility is a priority, a meaningful priority for people with Parkinson's. It's feasible to assess real-world mobility, and it's reliable, valid and acceptable. I've already discussed this. But hopefully these will be the next generation digital measures that you are going to be using in your clinical trials, your research and in clinical care. And I really hope that these will be coming to a clinic and a research centre near you in the very near future rather than in the 10-year future. We would expect to see these being adopted more widely very soon. I'd like to thank the funders for Mobilise, the EU, our industry partners, and also acknowledge my wider group who, through all of their work, have contributed significantly, and our wider funders. Thank you very much.
Thank you for a great talk. Time for a quick question.
Talking about the different digital, in the past we used to have the watch sign where we had analog watches, and people would think that their watch was not working well because they needed to move, and they would take it to various watchmakers and say, my watch is not working. And but now we have a digital watch sign. So I had a couple of patients telling me that we tell them to exercise, and he goes for a walk with his wife, and they both have their Garmin watches. And then he would say to me, my Garmin watch shows I have taken less steps than my wife, although we both walk the same park together. And it appears that the number of steps you do, as you rightly pointed out, is measured by the arm swing. So if you're not putting GPS on to know how much distance you've done, you can get an error in the number of steps they've done because they're not swinging their arms. So this is the modern Garmin sign of, like the analog before.
Very good.
Okay, thanks very much, Lynn. Thanks for a great talk and lots of more.
So I am delighted to welcome you back from a delicious lunch. Thank you to Sonia and the organisers for the afternoon session on therapeutics and innovation. Moving on to our third talk, I'm delighted to introduce Professor Tom Foltynie. He's Professor of Neurology here at the UCL Movement Disorders Centre and a consultant neurologist at the National Hospital for Neurology and Neurosurgery here at Queen Square. You're the chief investigator for a series of disease-modifying trials in Parkinson's, and especially of the EJS ACT multi-arm, multi-stage trial. So hopefully you're going to tell us about that now.
So thank you for the invitation. I'm delighted to be here, and I'm obviously just reeling from the fear of being replaced as a DVS neurologist by a robot and computer, which is looking imminently real.
I like it.
So I'm going to talk about EJS ACT-PD. So many of you will have heard about this, and if you haven't, it's because we haven't been doing our marketing communications very well. But the idea is that this is a revolution of clinical trial design to try and develop more efficiently or to evaluate potential disease-modifying treatments for Parkinson's disease. So we met with Max Palmer in approximately 2018. He'd been doing trials, platform trials, to look for agents which could improve the outlook of people with prostate cancer for about 20 years. And as a result of that meeting, a platform trial was set up for multiple sclerosis, for motor neuron disease, and it was the inspiration that we should be doing a platform trial for Parkinson's disease. Let me just, there we go. And so there, of course, are many issues in Parkinson's disease which are completely different to cancer and other neurodegenerative diseases, and there were lots of decisions we had to make. And we were very lucky that we managed to get funding from the Edmund J. Safra Foundation in order to address the challenges that would emerge. Now, of course, this is our conventional wisdom, that you have a setup process which is inevitable, and then you compare your potential treatment against placebo. There's a period of time to recruit, there's a period of time to follow up, and it takes some time, but at the end of that, you get your results. The trouble is that with that result, it usually doesn't end up with a licensed drug. It usually ends up with some plans to do another trial. And then you have to repeat the cycle and set up the process once again, which is hugely inefficient. You get a sort of preliminary result for one drug only. And we wanted to do better than that. So we had funding in 2021. And over the course of the next 4 years, we managed to address all of the issues that arise in order to get a fit-for-purpose disease-modifying trial that could test multiple treatments simultaneously and not just do a phase 2, but also collect data that was phase 3, fit for licensing and regulatory approval. So a huge amount of Zoom meetings, but fortunately, after the pandemic, we managed to have some face-to-face meetings as well. In order to achieve this, we involved all of the major stakeholders in Parkinson's research. So we set up a treatment selection working group that Tony Schapira led. We had an infrastructure working group thinking about all of the different sites around the UK, a trial design working group that Roger and James Carpenter led for us, an outcome measures working group that Annette Schrag was leading, and a funding working group, because of course this is a very ambitious project, and Sonia and Joy Duffin were led the way for that. But at the centre of all of this, we wanted to make sure that everything we did, all the decisions we made, were driven by and included the voice of patients. So our trial is to be inclusive. We want to look for disease-modifying treatments that have relevance for the broader population of patients with Parkinson's disease. So you can see, having a clinical diagnosis, as long as you're diagnosed over the age of 30 and you're on dopaminergic treatment, you're eligible. The exceptions are if you have atypical forms, if you have a very young onset genetic form, the Parkin Pink1 DJ1, or if you have an advanced therapy already, a pump or DBS. And originally we thought, well, we want to be representative of people with Parkinson's in the UK. We've kind of since realised that that's very difficult because we don't know what representative really is. So we're kind of changing that. We want to be inclusive and remove the barriers for participation that previously exist. Now, of course, everyone will immediately think, well, hang on, Parkinson's is hugely heterogeneous. For example, you can look at the genetics of Parkinson's disease, and this data from PD Generation, approximately 12.5% of Parkinson's patients have a single gene, mainly GBA1 or LRRK2, that you'll have heard about this morning already. But the vast majority outside those single gene disorders, 87%, do not have a single gene accounting for their Parkinson's disease, and it's that group that we really want to try and find treatments for. So we're collaborating very closely with GP2 and PD Frontline. So PD Frontline will look at the LRRK2 and GBA1 status of all of our participants, and GP2 will do whole genome sequencing for all of our participants. And so that means that when we have our trial results, we can not only know about the Mendelian, but we can also look at the polygenic risk scores. So polygenic risk scores that are relevant for the diagnosis of Parkinson's disease, but can also tell you whether you have a mitochondrial polygenic risk score, a lysosomal polygenic risk score, a neuroinflammatory polygenic risk score, which means all of our participants will be able to contribute what the predominant mechanisms leading to their Parkinson's disease have been. And this is going to be vitally important. Another issue in terms of inclusivity, of course, we're wanting to remove barriers in terms of age, remove barriers in terms of sex, remove barriers in terms of deprivation. And a huge elephant in the room is that many people that get involved in Parkinson's research tend to be white Caucasian, tend to be educated. And so there's a lot of work that we've got specific funding for to address the reasons why people from different ethnic backgrounds do not currently participate in these trials. And so thinking about trust, communication, and emphasising the importance of this project. So traditionally, if you do a trial looking for disease-modifying effects of a drug, you look, for example, the time it takes to levodopa, or you look at the MDS-UPDRS Part 3, which is the clinician's assessment of motor severity, or you look to see how long it takes for that score to worsen. So Cristina Gonzalez-Robles led some work looking at all of the different outcome measures that have been used, and has been pivotal in trying to think about how we take the patient view and emphasise that as our primary outcome. And that aligns with where all the regulators are moving. The FDA, the EMA and MHRA are all also prioritising outcome measures that are relevant to patients rather than just to clinicians. How quick is your finger taps, or how quickly can you move your hands? So that we've published a nice paper looking at how we can embed the patient voice amidst all of the different outcome measures that you could choose. And we use the critical path data set. So this is over 9,000 people who have contributed to previous studies, either observational cohorts or clinical trials. And then, of course, you can use that data to see how measures change over time at different stages of disease, at different ages, and across different genders. And with our patient group, we decided to select the UPDRS Parts 1 and Part 2 in combination as our primary outcome measure. And that has never been used before. But of course, this is capturing the non-motor symptoms of Parkinson's and the function, the activities of daily living of Parkinson's disease. And using the CPP data set, we've been able to look to see how this changes over time to make sure that this is a valid way of assessing Parkinson's getting worse. So this is statistically valid, it's sensitive to change, and allows us to calculate the sample sizes that we need. What's more, by using this Part 1 and Part 2, this patient report, means that we can have a trial design that means that people who live a long way away from a trial centre can still participate. In the era, the post-pandemic era, where everyone can be on Zoom and Teams, it's perfectly possible to collect all the information you need remotely, as long as every participant has their first visit face-to-face to ensure safety. On top of that, we've wanted to make sure that we have direct-to-patient dispensing, and so there's no need for a participant to come to a centre to collect their medication. Now, we've worked out as a result of this primary outcome, the Part 1 and Part 2, that it takes 3 years for there to be substantial worsening over time, because fortunately Parkinson's disease is a slowly progressive disorder. But of course, that is frustrating. We want to have signals that can predict worsening much more sensitively. And that means we're using novel exploratory outcome measures. For example, the activity lumbar sensor, the size of a postage stamp, which will be fixed to a participant's lumbar spine for a week, every 6 months throughout the course of the trial. We'll be taking blood samples from people and plasma and serum stored to look for wet biomarker changes over time that might be able to predict our primary outcome. And in subsets, there will be brain imaging to look for structural changes at a micro scale. So this is how the overall trial design looks. Our treatment selection working group advised us that telmisartan and terazosin were very strong candidates, and indeed they are. And those are the first 2 treatment arms that will be used. Everybody has standard of care, and those are once daily preparations that will be compared with placebo tablets and standard of care over time. In September this year, we'll add a 3rd active treatment arm, which is UDCA. Now, UDCA has got a different dosing regimen. It has to be 5 tablets split between 3 doses during the day. And so we can't compare that directly with a once daily placebo regimen. You know, it's possible that there's a minimal difference, but it's possible that there might be a difference. And so we're going to introduce a placebo 3 times daily regimen and follow that placebo 3 times daily in comparison with a placebo once daily to ensure that it is valid to combine these and regain the efficiency that we need. So we'll be recruiting 400 people to each of these treatment arms. This is a huge, ambitious project. Everybody will be on their randomised allocation for 36 months. So we're looking for a total of 2,000 people in total. And we will have enough power to detect an effect of 30% or more in the rate of progression in the Part 1 and Part 2 combined. And we'll be doing interim stages. You can think of these interim stages as being the phase 2 outcome. So if a if a drug looks no good, then we will stop it and we will replace it with the next best candidate. Whereas if a drug passes these interim stages, rather than having to stop and reset up a new trial, we'll continue following up the existing patients, recruiting more patients until we have a definitive phase 3 outcome at the end. So all of our patients will get the digital and the sensor on the lumbar spine. All of our patients will be genotyped to enable these polygenic risk scores, and everybody will have a wet plasma and serum taken at the start and at the end. At some sites, particularly those where people are coming for face-to-face visits, we'll have additional lumbar punctures. There'll be interim blood tests looking for early signals of change, and as I said, the MRI imaging. So the ultimate bar is, of course, to find drugs that slow down disease progression. But the earlier interim low-hanging fruit, if you like, the benefits that we'll realise is that we'll be setting up over 40 sites across the UK where there will be trained staff in place, and we have a training regime set up, and they will be therefore able to deliver trials, not just ours, but other trials in Parkinson's disease. This is creating an infrastructure that will help other commercial or academic trials to be performed. We've developed a recruitment infrastructure, and I'll tell you on the next slide about the Joint Parkinson's Research Registry and communication infrastructure to keep all of our participants and people on the waiting list informed. We'll have expert staff delivering this trial in one person at least in each of the regional research delivery network regions across the UK, and those core funded staff will support people within their region and share knowledge and best practice and make sure that recruitment locally is inclusive, because there'll be different things which prevent recruitment or inclusivity at different sites. And we're fostering a cohort of early career researchers to make sure that, beyond our immediate involvement, this is a long-term project that's going to last potentially for decades, and we'll need people to take this on. So the Joint Parkinson's Research is part of Research Plus Me. There's a QR code, and it means that people who are interested in Parkinson's research can sign up for this. There's no gatekeeping by their Parkinson's clinician. People can go direct to this, look at all of the different research opportunities for Parkinson's across the UK, and then if they're interested in our trial, they can sign up for our trial directly. And that is empowering, and that helps the inclusivity, that it's not just the initiative of the treating physician to signpost people. So we launched last September. This is the largest Parkinson's disease trial. Of course, we've had lots of advertising for this, and this is why I hope that people in the room are not hearing about this for the first time. So this was last October. We had news outlets, various publications in multiple countries have picked this up, and we had 800 people sign up to join this trial, their registration of interest, using this online platform within the first 48 hours. Since then, of course, as part of that, we've got more than 100 people in our consortium who have helped design this project from 54 different institutions. We've had 28 people who are either patients or their partners helped contribute to this design. We have 11 early career researchers. We've had more than 50 abstracts at conferences. We've published 10 papers, and this has been spread over 10 different countries. And we've had more than £30 million worth of support for this. So this is a huge, ambitious project that has to change the face of how we are delivering Parkinson's trials in the UK. Up to date, we now have over 1,800 people who have signed up the registration of interest. Of course, this is a way of pre-screening and making sure that people are eligible so they don't then waste their time coming to sites. 92% are white, 8% are from other ethnic groups. That's a little bit short of our target. We should be nearer 10% from minority groups. There's quite a good gender split, and there's quite a reasonable age distribution as well. And all 4 of the devolved nations will have sites recruiting within them. So as things stand, we have 16 sites that are currently open. and those are the green dots on the map there. The orange and blue sites will be opened, and we're putting constant pressure on those sites to try and get the paperwork done so we can get the green light and get them open. So as things stand, we have 114 people that have been screened. A few have not been eligible despite the pre-screening that can take place, and this is for things like blood pressure and other things that can only be detected face-to-face. We've got 88 people who have been randomised, and so far we've had 5 withdrawals, and so it's a percentage, but fortunately it's only quite a small percentage, and that's bearing in mind that we're using drugs which lower blood pressure, which of course can be an issue with some Parkinson's patients. So looking forwards, we will open our 3rd active treatment, UDCA, in September this year. Chris Lambert is leading our imaging sub-study, so we'll have an hour's worth of MRI data, all of the relevant sequences, looking at structural change over time. We are already considering what the 4th, 5th and 6th treatment arms will need to be, because there's a long time between making that decision and getting the funding and then the manufacture and then the delivery at site. So even though we're not expecting to need our 4th, 5th and 6th treatment arms for a couple of years, we have to get all those decisions made now. And of course we're collaborating both within the UK and internationally with other partners, and that's particularly relevant here. So of course we're at the top there, we're a phase 3 platform trial, and that's our primary outcome measure. And we are working very closely with colleagues in Norway who are doing another phase 3 equivalent trial called Hydra. Our French colleagues are doing the NS-Spark master trial, and our colleagues in the United States are doing a prodromal trial called Pathway to Prevention P2P. So as a group, we formed a global alliance of Parkinson's platforms, so we can communicate directly and know what drugs people are testing, what lessons they're learning in terms of what helps recruitment, and how well different outcome measures and exploratory measures are performing. What we really want to plan for is what we get at the end. So we're going to have a huge clinical data set, and it'll be the clinical data set which dictates whether drugs are fit for purpose or not. But we're also going to have a huge amount of digital, genetic and omic data from the samples that we have, both plasma serum and CSF. We're going to have a huge data set which lends itself to all forms of artificial intelligent multi-omic factor analyses and the convolutional neural networks in order to disentangle signals that we don't yet fully understand. So this is hugely exciting. Very happy that as a result of our patient involvement and the team that we've got working on this, that we were finalists in the Made with Patients Awards, and that's very much celebrating what is at the core of this trial, that we want to be doing things which are relevant to patients. So we have a QR code for people that want to participate, that there's no gatekeeping, people that want to register can do so. And we also have a QR code for commercial companies, because the long-term sustainability of this platform is not going to be long-term charity support or government support. That's what we've currently got. But we need to make this appealing to the commercial sector so they buy in and use this infrastructure to test their drugs in the UK. Thank you very much for your attention.
Thank you, Tom. We've gone from one frontier to another. And I think there's a few things that are really inspiring me. I mean, firstly, disease-modifying treatments in patients. I love the platforms talking to the platform, so a platform, and the patients at the heart of it. And I think that's really, that's one of the things that's been so successful, that you've really had right from the beginning. And that's what's inspired me personally about working on this. That at every one of your working groups, you had 2 people with Parkinson's involved. And I think that's why this is so successful. So, any questions? Roger, perfect.
I'll just shout.
Yeah.
The 5 who dropped out, what was that because they found it too intense? Was it blood pressure problems? Because it's about 5 or 6%, so it could be quite sizable.
Yeah, so we have powered to have 30% dropout over the course of the 3 years. So we're anticipating some people won't make it to the end. Of those 5, yes, a couple are because of blood pressure. One has been a potentially unrelated side effect, but she's attributed it to the drug and therefore wanted to stop. And at least one has been they just didn't like the idea of the trial visits.
Great, thank you. Thanks very much. So we're going to maybe everyone stretch for a bit because we've had so much innovation, and now we've got innovation in the, I want to say the next generation, but they're not that much younger than me. So I'm absolutely thrilled to introduce Ed Jabbari, who is a research group leader at the UCL Queen Square Institute of Neurology and consultant neurologist at the Movement Disorder Centre. His research really integrates fluid biomarkers and genetic approaches to enhance diagnostic accuracy for Parkinsonian disorders and to refine clinical trials. And I can see that they will absolutely feed into the sorts of trials that we've been hearing, and to discover novel therapeutic targets. You were recently awarded an MRC Clinician Scientist Fellowship to establish your own group at UCL. So hopefully we're going to hear a little bit about that.
Perfect. Thank you, Ramona, for that very kind introduction. So I'm going to give a kind of high-level summary of some of the translational work that we're doing in my very newly established research group here at the MDC. And in a nutshell, what we do is, as Ramona's already said, we integrate genetic and fluid biomarker approaches to address the major unmet needs across Parkinsonian disorders, in particular Parkinson's disease and PSP. So when I'm referring to these major unmet needs, what I'm referring to is that in the absence of objective diagnostic biomarkers, we run into issues of clinical misdiagnosis and delays in diagnosis. And of course, this feeds directly into clinical trials because we want the right patient in the right clinical trial at the right stage of disease. The other issue, of course, is predicting the rate of clinical decline, which we know is highly variable in both Parkinson's disease and PSP. And the reason why this is important from a trials perspective is because we want homogeneous clinical trial cohorts that have similar disease trajectory profiles. So this is a really important unmet need. And then the obvious unmet needs. We need effective disease-modifying therapies, and we need innovative clinical trial design. And in my allotted 12 minutes, I'm going to try and tell you how we are tackling each of these issues. So the first one is achieving early and accurate diagnosis and prognosis for our patients. And this really speaks to our work in the seed amplification assays. So I'll give you my quick crash course in how these assays work. Essentially, we are using these assays to capitalize on the self-templating properties of pathologically misfolded proteins that define these various Parkinsonian disorders. So alpha-synuclein for Parkinson's disease, 4-repeat tau for PSP. So essentially, if you take your patient's biofluid sample, you mix it with a recombinant form of that particular protein that you're trying to detect. If your patient's fluid sample contains those pathologically misfolded seeds, it will induce the misfolding of the recombinant form of the protein, leading to aggregate formation. And through the addition of a fluorescent dye called thioflavin T, you get these nice aggregation curves where you can detect and track the aggregation. Now, the landmark paper that really put this assay on the map was the PPMI study, the Parkinson's Progression Markers Initiative study, where the application of a commercial alpha-synuclein assay showed that the assay had high sensitivity and specificity at differentiating Parkinson's from healthy controls. The field then evolved and considered this assay not just as a binary positive or negative outcome, but also explored whether these quantitative seeding kinetic measures that you can derive from the aggregation curve, like the lag phase and the maximum fluorescence signal, whether these had diagnostic value at differentiating between different alpha-synuclein strains or conformers. In this case, differentiating between Parkinson's disease versus MSA type seeding. So clearly there is diagnostic value in this assay. No one is doubting that. However, at this point, certain parts of the world, in particular our collaborators and our colleagues from the US, apologies if anyone here is from the US, were slightly, shall we say, overzealous about the application of this assay, and they were saying this is ready for prime time, let's roll it out into the clinic. And the scenarios that they envisaged was that you'd have an early Parkinsonian patient, you're not sure whether they've got bog standard Parkinson's or one of the atypical syndromes. Let's run this test. If it's positive, they've got PD or MSA. If it's negative, they've probably got PSP or CBD or something else. Of course, we know that that's way too simplistic because of this concept of co-pathologies, which are the rule and not the exception across neurodegenerative diseases. This was a really simple study that we did to highlight this point. We took our PSP and our CBS CSF samples, we applied the commercial alpha-synuclein SAA to those samples. 20% of those samples were alpha-synuclein SAA positive. This should come as no surprise because we know that at post-mortem, up to 20% of PSP patients have Lewy body co-pathology, and that's what this assay positivity reflects. So the lesson that we've learned from this is that, again, as we already knew, there is no one perfect biomarker, and we can't use the alpha-synuclein assay in isolation. The model that we have proposed that we want to move towards is a combined panel SAA approach, where we're taking a granular approach to molecular diagnosis, multiple different protein pathologies for enhanced diagnostic accuracy. The first step towards this, of course, had to be the development of a 4-repeat tau SAA. And I've shown this many times before. This will be very boring for people who have seen this a lot, but we've started with developing the 4-repeat tau SAA in post-mortem brain tissue. This was in collaboration with the NIH Rocky Mountain Laboratories group. You can see 6 PSP brains going from top to bottom from Queen Square Brain Bank. And in all 4 technical replicates in each sample, we achieved assay positivity. We've then spent the last 12 months optimizing this assay to work in ante-mortem CSF from living patients. And we are now starting to see reproducibility in this assay, picking up positivity in PSP patients, but achieving, as we want, negative results in Alzheimer's being a mixed 3 and 4 repeat tauopathy, and in Parkinson's disease. And again, this is in collaboration with the NIH group and also Jarko Compter from Barcelona, who's here in the audience today. So we think that this is going to be a real game changer in terms of that combined SAA approach. And I'm going to come back to this in a minute to talk about the big picture and what the next steps are. I'm going to briefly segue now to disease progression and prognosis. So if you remember a few minutes ago, I told you about these quantitative seeding kinetic measures that you can derive from the aggregation curve and how they may have diagnostic value. The question that we wanted to answer in this study was, is there prognostic value in those kinetic measures? So if you take those measures at baseline and you follow patients up over time, can it predict clinical decline? We answered this question in over 1,000 PD participants, a majority of them from PPMI, and with some independent replication cohorts as well. And quite convincingly, what we found was that the speed of the seeding kinetics, which is the lag phase or the time to threshold, as it's also known, the faster that time to threshold, the much higher the probability of going on to develop cognitive decline and Parkinson's disease dementia during that follow-up period. So what we think mechanistically how this is explained is that the faster the seeding kinetics, the more aggressive those kinetics, much more likely you are to see cortical spread of Lewy body pathology leading to cognitive decline. So we think this is now adding a prognostic arm to the assay as well as the diagnostic side, which we already knew about. How is this all coming together at UCL? So through MRC fellowship funding, I've recently set up my research group here at the MDC, and the approach that we're going to take is exactly what I've alluded to. Combined panel SAA testing, initially starting with synuclein and 4-repeat tau for this granular molecular diagnosis. There are, of course, as you can imagine, also plans to eventually progress things and roll this out as part of a clinical diagnostic service here at Queen Square. And there is a real team effort to get this going. So between our group, also other members from the MDC, in particular Sonia, and also people from the UK DRI, in particular Tim Bartels and Yen Go. And we will eventually get there. So, and Nishant Vaiketh, who's recently joined my group as a very experienced senior research fellow, is really spearheading the setup of these assays in our lab, and we are actively testing samples as we speak. What's driving this also is our new UK-wide longitudinal cohort of early Parkinsonian patients. And this really is the perfect type of cohort, this clinical grey area of early stage Parkinsonism for diagnostic biomarker development. So I'm going to briefly talk about disease-modifying targets with a particular focus on LRRK2. Our interest in LRRK2 began around 5 years ago when we conducted this study. This was a survival GWAS in PSP. And to our surprise, we found a genome-wide significant hit on chromosome 12. And when we dug a bit deeper, we found that this related to the LRRK2 locus, and these lead SNPs that were determining faster progression were also determining a much higher expression of LRRK2. So this raises the question, is LRRK2 a disease-modifying target in PSP? So spurred on by this, we obtained some postdoctoral research funds from Cure PSP, and we conducted this study that we've just recently published in Movement Disorders. The take-home message here, this was in collaboration with Esther Samler from the University of Dundee, who has developed translational blood-based assays that can quantify the levels of LRRK2 and LRRK2 kinase activity. And the take-home message here is that the same direction of effect that we saw in the GWAS, the higher the levels of monocyte LRRK2, the more rapid your clinical decline over the course of 1 year. So understandably, this has generated a lot of interest from the drug companies that are developing LRRK2 therapeutic programs, and we're in active conversations with them about whether there's potential to set up a LRRK2 trial for PSP patients. So watch this space, there'll hopefully be more on that soon. What about LRRK2 in Parkinson's disease? Surely this is more straightforward. LRRK2 PD patients, surely that's a homogeneous group of patients that you can study in a clinical trial. We know that that is definitely not the case, because 30 to 40% of LRRK2 PD patients are alpha-synuclein SAA negative. And this mirrors what we find at post-mortem, where up to 50% of LRRK2 PD brains are Lewy body negative. So this begs the question, are there biological subtypes within LRRK2 PD? And is there an alternative primary pathology that's driving this subset of LRRK2 PD patients? And now what we have at our disposal is a really powerful tool in the SAA at probing that. What we're doing actively as we speak is we are applying both our alpha-synuclein and our 4-repeat tau SAA across multiple brain regions from LRRK2 PD brains that are in Queen Square Brain Bank. And this is very preliminary data, but we are seeing evidence of 4-repeat tau seeding in a subset of LRRK2 PD patients. So this really speaks to this hypothesis that there could be biological subtypes within LRRK2 PD, and perhaps tau is a therapeutic target for that subgroup of patients. For the last 90 seconds, I'm not running over, am I? No, good. Tom's already introduced the concept of the need for innovative clinical trial design for both Parkinson's disease and for PSP. As we've already heard, the way these multi-arm, multi-stage clinical trials work is they trial multiple drugs against a common placebo group, so much more efficient in testing potential disease-modifying therapies. MND SMART paved the way for Octopus in progressive MS, and now most recently, the EJS ACT-PD trial. What we're very interested in is whether SAA could have a role in these trials. Now, the obvious thing would be to look at SAA status, our panel SAA approach of positive or negative, to be used as inclusion or exclusion criteria for the trial. And based on the data that I've shown you for the SAA kinetics, it's also possible that these could be used as a stratification tool and also as markers of target engagement and treatment response. So again, in collaboration with Tom and Sonia, this is something that we'll be able to look at in the EJS ACT-PD trial. Finally, I'm also really pleased to share that We have made very good progress in the setup of the PSP multi-arm, multi-stage clinical trial. The wheels are very much in motion, and we'll hopefully be able to launch this at some point next year. And of course, our 4-repeat tau SAA is going to be embedded in the clinical trial design for PSP Smart. So to conclude, we're using combined SAA testing for early and accurate diagnosis and prognosis across Parkinsonian disorders. LRRK2 is emerging as a promising therapeutic target for PSP. Tau may be the primary pathology in a subset of these LRRK2 PD patients. Is it a therapeutic target? And finally, we've seen the benefits of innovative trial design for Parkinson's disease, and we're going to be launching the same for PSP. And thank you very much for listening.
I think we'll go straight to the next speaker because
of course.
I want to make sure we get to coffee, and maybe, I don't know if there are biscuits, we'll find out. So, Patrick, yes, wonderful. So I'm delighted to introduce Dr. Patrick Cullinane, who's going to wrap up our innovation session. Patrick is a neurologist and the current Edmund J. Safra Clinical Research Fellow at the UCL Queen Square Institute of Neurology, and his research focuses on defining the biological basis of clinico-pathological diversity in Parkinsonian disorders. So, over to you, Patrick.
Yeah, so thanks very much for the chance to present our work. I'm going to discuss today how we're using the Queen Square Brain Bank cohorts to investigate the biological basis of clinical pathological heterogeneity in Parkinsonian disorders with the long-term aim of improving patient stratification and identifying targeted therapies. So as the Queen Square Brain Bank is turning 40 this year, I just wanted to highlight several incredible studies which really set the scene for the topics I'm going to discuss. And this ranges from early studies on Lewy bodies and aging, regional selective vulnerability in the substantia nigra and PD, to the transmission of Lewy bodies to fetal grafts, supporting the prion-like hypothesis of spreading. And then more recently, studies showing G51 mutation in the synuclein gene produce synucleinopathies with pathological features of both Parkinson's disease and multiple system atrophy. And the Queen Square Brain Bank has also provided tissues for many landmark studies, including the recent discovery of the cryo-EM structure of alpha-synuclein filaments and large-scale visualization of alpha-synuclein oligomers across the brain. So collectively, these studies have shaped our understanding of Lewy pathology, concepts such as selective regional vulnerability, clinical pathological heterogeneity, and driven by technological advances, molecular mechanisms as well. But traditional clinical pathological studies have important limitations. Cohort sizes are typically small, the depth of clinical information available can be limited, and the semi-quantitative methods for measuring pathology burden as well. So we have over 3,000 brains in the Queen Square Brain Bank, and with advances in AI and digital pathology, I think we're entering the next generation of clinical pathological studies. We can now use machine learning-based methods to quantify pathologies across all brain regions at scale. And we can integrate existing molecular data on these cases, such as whole genome sequencing, to perform data-driven subtyping of patients. But in order to interpret these subtypes, we need comprehensive clinical information on each patient. And using AI to extract clinical information from records, we can reconstruct very rich clinical information on patients throughout their whole life, which then we can use to further refine these data-driven models. And ultimately, we can use this to better select cases and tissues for downstream molecular profiling. And I guess ultimately, this will allow us to integrate detailed clinical, quantitative pathology, and targeted molecular data with the goal of identifying biomarkers and therapeutic targets that we can then test or validate in donor-derived experimental models. So in the few minutes I have, I'm going to give some concrete examples of how we have been progressing each of these elements. So I work very closely with Sonia, neuropathologist, whose team have developed quantitative pipelines for measuring regional pathology burden. And these have been used to quantify alpha-synuclein, tau, amyloid beta across cohorts. And we applied this to 236 PSP cases, mapping tau pathology burden across 42 brain regions, and this corresponded quite well with clinical phenotype. We also gave this data to machine learning models, and this identified 3 data-driven subtypes with distinct progression patterns. But we also found in PSP that the regional burden of tau pathology only partially explains disease progression. So we need to look beyond the primary pathologies, as others have mentioned, and think about co-pathologies. And of course, in the case of PSP, this means Alzheimer's co-pathology, Lewy pathology, and TDP-43 proteinopathy. But there are also co-pathologies beyond the kind of canonical misfolded proteins, including lymphocytic co-pathology. And again, we developed a machine learning algorithm to quantify T-cell burden in brain tissue. And we've applied this to the substantia nigra and cerebral peduncle across a range of conditions. And we found that in the substantia nigra, T-cells are higher in PSP and TDP-43 compared to controls. But I guess this doesn't really answer whether T-cell presence in tissues is a cause or a consequence or even a synergistic factor in neurodegeneration. So we're expanding this work to try and get a clearer picture of this. In addition, there's vascular co-pathology, including hyaline arteriolosclerosis. Again, we're trying to measure this quantitatively at the moment, manually measuring vessel wall thickness, perivascular space depth, and our early preliminary data shows that there is differences across conditions. And at the moment, we're developing deep learning models to really scale this measurement up. And then, as Ed mentioned, differences in strains of pathological proteins may also explain differences in clinical phenotype and progression. I'm going to kind of discuss this in the context of this case. So this macroscopic image and histology are from a patient who had frontotemporal lobe degeneration with synuclein. And this case is unusual in that it had features of both multiple system atrophy and Parkinson's disease pathologically. So we suspected a mutation in alpha-synuclein genes such as G51D, but sequencing was negative for that. So then we took filaments from this case and we seeded them in biosensor assays. And the seeding of this case looked exactly like typical MSA. And then we went on and got the cryo-EM structure of filaments from this case, which were again exactly like typical MSA. So I think these experiments kind of demonstrated that this is a rare pathological subtype of MSA. But then at the same time, we also seeded alpha-synuclein filaments from G51D cases, and that looks exactly like the seeding pattern you get when you seed material from Lewy body cases. So we kind of hypothesized at the time that the cryo-EM structure of G51D cases would look like the Lewy fold, and this structure has recently been solved, and it actually has features of both the Lewy fold and the MSA fold, which again, is exactly what we see in the pathology. And I've recently received funding to continue working with structural biologists to kind of scale cryo-EM, which is usually just used on a few cases, to look at larger case series of MSA with the aim of understanding whether conformational differences actually drive clinical diversity. And then we also have to think about the cellular drivers of regional vulnerability. We have identified that in patients with PSP and the corticobasal phenotype, they not only have higher amounts of tau pathology burden in the cortical grey matter of the precentral gyrus and parietal lobules, but also in the subcortical white matter. And in some cases, you can see this ribbon of dense tau pathology in the subcortical U fibers. So we looked at this region, GFAP positive astrocytes and P2R12 microglia, using spatial transcriptomics. And what we found is that there's a phenotype, region, and cell-specific expression signature in CBS that we don't see in Richardson syndrome or PSP Parkinsonian subtypes. And the astrocyte expression signature in this region is really one of exhausted astrocytes and upregulated inflammatory pathways. So we are now expanding the spatial transcriptomic work in PSP with the aim of transforming our data-driven progression models from a descriptive model to a more mechanistic model. And then how do we get the large-scale clinical information that we need to give to these models? So in the Queen's Square Brain Bank, we have around estimated 500,000 pages of clinical documents in the archives, and we're using two pipelines. So the first is we take the scanned PDF documents, we apply optical character recognition, and this extracts the text, which we can then give to large language models for clinical information extraction. But we can also give the PDF images straight to a vision language model to do the extraction. And using this approach, we can go beyond the kind of demographic, clinical phenotype, progression milestone type data that's traditionally been used in clinical pathological work. And we can now really collect much larger volumes of data on patients' medical histories, including vascular comorbidity and lifetime blood pressure control, which we can then compare back to the pathology to interpret what we found on that side. We can also collect information on all of their healthcare-related encounters, whether they attend a subspecialist clinic or general neurology, socioeconomic factors, occupation, whether they lived most of their life rural or urban setting, as mentioned, lifelong blood pressure, and all of their lab indices over time. And this is just an example of one of the pipelines. So we take the medical records that have been scanned, we give them to a light vision language model, and this parses the pages out into handwritten or typed. And it also looks to see what pages might have clinically useful information. And then those pages are brought forward to a much larger vision language model with 32 billion parameter training, and that does the actual extraction of clinical features. And then this is just one example of how we're doing it to extract blood pressure and the exact date of the clinic or when the blood pressure was taken, and aspirin therapy. And we looked at this in 1,000 pages, and we are getting accuracy levels compared to a human generated ground truth of about 93%. And in terms of the computational demands, to run 1,000 pages through this pipeline takes about 50 minutes on a high-end GPU. And just very, very briefly, I want to mention work we're doing in parallel on translational pipelines. So we're developing iPSC-derived and directly reprogrammed neurons from fibroblasts taken from QSP brain donors. And then we plan to use this model to seed alpha-synuclein filaments taken from the brains of the same donors. And we can also use this platform to validate therapeutic targets from our omics studies. And very last thing, we are adopting new technologies, including nanopore sensing of tau and alpha-synuclein post-translation modification as a potential future biomarker for neurodegeneration. And that's it. Thanks very much.
Thank you. Well, you've really shown us how you're going from the wet lab and then bringing it right through into patient care. So thank you. What a terrific session. I mean, my mind is blown.
Okay, welcome back, everybody. I really hope that you have enjoyed the day so far and are enjoying some of the breadth and depth of what we're trying to present here, right from the sort of molecular level diagnosis through to what can be achieved in terms of measuring patients and their movements, but also what can be achieved through implantation into the brain, which was super exciting. But now on to arguably the most important session of the day, and one that I'm really looking forward to. So our guest today, Rory Cellan-Jones, really needs no introduction, but of course spent 40 years with the BBC, and during that time as technology correspondent, really reporting on rise of the internet, the digital revolution, and technologies that really shape how we live our world. But of course, in recent years, Rory's perspective has shifted from observing technology at a distance to now experiencing what it's like to use technologies for health with their benefits and their problems. But what makes Rory's voice, of course, very valuable for you as an audience is that he sits at these rare crossroads of really understanding both technology as well as a condition. So we're going to explore his journey just for maybe 15, 20 minutes or so, and then I'll open it up to questions from all of you. So, Rory, first of all, thank you very much for being here with us.
Well, great to be here. And I've sat in awe all day. Struggled sometimes with the science as somebody who gave up biology at 12, I think it was, but been absolutely gripped, and also felt honoured to be. I'm on the EJS ACT 100 trial, so
fantastic.
Very excited about that.
Great. I'm not sure I've interviewed a participant yet, so thank you very much. Okay, well, just going back to your days at the BBC, maybe to tell us a little bit about, you spent those decades explaining technology to millions of people. What drew you to that whole world?
Well, again, I'm a complete fraud. I did a degree in modern and medieval languages at Cambridge University, and my first real encounter with a computer was to walk into the BBC newsroom where I was a young producer in 1984, I think, and promptly go out on strike over computers, because they were bringing in computers to replace this fleet of ladies who typed up the scripts, and our union told us we had to go out on strike over this because this was a terrible, terrible thing. And actually, the real story was that ITN, who are always much richer than us, had paid each of their journalists £1,000 a head to accept the horrible computers, and we, in the end, settled for 300 quid. So that was an unfortunate first encounter. But what happened to me was I was a business correspondent for a while. But by far the most interesting business story in the late 1990s was the rise of the internet. And we got our first family computer in 1996. And I lived next door to a medical illustrator. She said, "You've got to have a Mac." We knew nothing about computers. I bought for 1,500 quid a Performer 630 with a 250 megabyte hard disk, and I bought a modem for another 250 quid, and plugged it into the wall. And the computer first came on and played a little cartoon. It was me, my wife, and our then 5-year-old son just gathered around this little box. And then I managed to find out how to go to a website. And the website was the Louvre website, which was one of the very first websites. And onto the screen, line by line, animated a picture from the Louvre. And it was, I forget who said this, it's like all great technology, it appears like magic at first. And I was hooked by this, and gradually managed to inveigle my way from being a business correspondent to being a technology correspondent.
Wow, those days. I was watching Young Sheldon with one of my daughters, and they had a scene where they had a modem. And when I grew up, so modems had a very specific noise when they connected to the internet. Yeah, and there's this moment, and none of my children understood at all why this was so humorous to me. So I mean, you did this. Obviously, you became technology correspondent for a long while, and then tell us about the time your diagnosis was made and how that happened, because it was in a very public way in the end.
Yeah. I mean, I was technology correspondent officially from 2007. Started my very first story officially, although I'd been doing it for most of the previous 10 years. My first official story, by the way, was to be there in the hall when Steve Jobs unveiled the iPhone.
Oh, wow.
And just a little story there. I knew that was going to be a huge story because as we legged it out of the hall, 8 hours behind, as it were, to try and get a piece on the 10 o'clock news, London rang, and the desk in London was never interested in technology stories, never gave a damn, but somebody was shouting, "You've got to have your hands on that phone!" And I said, that is impossible because Apple is the world's most controlling company, and phone's not coming out for another 6 months. But then I remembered that they'd promised an interview with, not Steve Jobs, but his tubby little marketing guy, deputy, Phil Schiller. And I'd said yes, but with no intention of doing it. And I changed my mind, raced back, said to Phil Schiller, "Have you got the phone?" "Oh, yes." Held up the phone, did a piece, and got it on the 10 o'clock news. And the next Sunday, the Observer's tech columnist, John Norton, said that I'd looked like a French medieval peasant a peasant holding a piece of the one true cross, which was true because it'd been such a desperate rush to get it. Anyway, I just had to get that in. So by 2019, when I was diagnosed in January 2019, 2 days before I flew off to Las Vegas for the annual Tech Fest. I didn't think my symptoms were that obvious. It was mainly a dragging of my right foot while we were away on holiday. But I did have a bit of a tremor. And somebody had written into the BBC before I was actually diagnosed saying that man ought to see a doctor because I'd been seen holding a cable on a beach in Jersey, talking about fibre broadband and my hand shaking. But in May, I covered the arrival of 5G in the UK with a live broadcast onto breakfast television. And after I'd done it, my producer said to me, and she knew, and sort of close friends and colleagues knew, have you ever thought of going public? And I said, yeah, I have thought about it, but why do you say in particular? She said, well, your tremor was very evident. And I had a look back at the pictures and I was all over the place. And so we were heading off on a train to Birmingham to film more about 5G. And at Birmingham International, I put out a tweet saying, some people have noticed my hand shaking, I've been diagnosed, I'm getting good treatment, onwards and upwards. And by Birmingham New Street, when I got off, my phone was melting with thousands of messages, yes. Which was, it was the good days. This was when Twitter was Twitter, not X, and it was a lovely place. It was 49,999 nice messages, and one man saying, "You've been standing too close to 5G phone masts."
Is that true?
Well, yeah.
Yeah, so you were outed, really. I'm not sure you had a choice in the end. And I guess, you know, after that diagnosis, and you're obviously very aware of your symptoms, as were all of the viewers, just how did your day-to-day life change?
Not radically at all. I mean, I had this sort of honeymoon period. Although what was strange was I had a couple of motor symptoms. I did have one non-motor symptom, which was depression on and off for the first year, which funnily enough dispersed when we went into lockdown because I was still working and I had a weekly radio show on the World Service, and instead of doing it from a studio, I was doing it from my loft to my producer's kitchen in St Albans and whatever, and it was a big new challenge and quite exciting, and that kind of brought me kind of out of my depression. And but for a while, I secretly thought, what's all the fuss about? Yeah, which may have been partly because I had faced a much bigger health challenge back in 2005 when I was diagnosed with a malignant melanoma behind my left eye, cancer, for which I'm still monitored. So which had seemed absolutely terrifying and imminent, whereas Parkinson's seemed, oh God, another bloody thing. So
slow.
Yeah.
And I mean, we've spoken before about this, but it's a common experience of people with lived experience is that they're always amazed that, well, for a condition like malignant melanoma, you'll have had a lot of tests and investigations, and whereas for something like Parkinson's, a clinician will have done
it's all vague,
due to do this.
Yeah.
And I mean, how does that feel? Not having sort of deep investigations and diagnostics for.
Well, it was kind of weird. I mean, I wasn't surprised by the 4-month wait between seeing my GP and seeing a specialist, but the specialist I saw was the registrar rather than the consultant herself, and it was kind of, oh, you're showing signs of Parkinsonism. Oh, really? That's not good. And you'll probably see the consultant, but it may be some months. And it was kind of, you what? And I actually made a fuss and got to see the consultant much quicker. But there seemed to be...
Were they any better?
Absolutely, much better. I mean, I had the loveliest... I mean, all the cliches were the wrong way around. My consultant was warm and cuddly. A lovely lady called Joanna Ball, who was very close to retirement. And I met my first Parkinson's nurse was a middle-aged man who made me walk up and down and said, "Oh, that looks bad." And I never went back to him. I've got a very good Parkinson's nurse now, but yeah.
So, I mean, just for the audience here, you know, it would be really helpful, especially the healthcare professionals in the audience. You know, what makes a difference? What do you call a good clinician? Somebody who...
Well, what I always say, rather sort of bitterly, to when I write about this, because I write a lot about communication in the health service, which is not brilliant. I say the world's most valuable thing, and the only profession that keeps it a secret is your consultant's email address. Yeah. I've now got a very nice guy at Charing Cross, Yen Tai, who I try not to email much. I try to email the Parkinson's nurse, but just being able to, you know, ask daft questions like, you know, why am I only sleeping 2 hours a night? Yeah. Is there anything that can be done? Yeah. Responsiveness, really.
Yes, okay. Even when, as you and I both know, a lot of the responses that we have are full of uncertainty because there's so much we still don't understand. So let's move now to what your views are, less on clinical and clinicians, but a bit more on the research, because you've also been so engaged with understanding, I know because you've come to my lab and seen the research I do, but just much more broadly, I think you went to World Parkinson's Congress recently, and you've been really engaged in following that research, both on tech but also on every aspect of Parkinson's. And I wondered what your view was from your perspective on how is that research going. And
well, what I've been doing for the last year nearly is working on a book about the quest for a cure and whether AI could make a big difference. And the book has turned out to be a little bit about AI, because it's based on Demis Hassabis' line that over the next 10 to 15 years, there's a good chance that AI will solve all diseases, which is a pretty big claim. But then he told me in 2020 when I interviewed him that he would do some Nobel Prize winning science in the next 5 years, and I thought, yeah, and then he won the Nobel Prize for chemistry. So he's to be taken seriously. But what I've found frustrating is the slow pace, and I kind of understand it. But what really strikes me is how far we still are from really understanding what's going on. I mean, you're doing all this great work in the brain. Others are doing, you know, hunting down mitochondria and whatever. But there doesn't seem to be I went to an extraordinary debate, actually. Well, I have to confess, I missed it, and somebody gave me a recording. I listened back to a debate with somebody in this room arguing that alpha-synuclein wasn't the be-all and end-all. It was an enthralling debate in Phoenix, and
I'm just going to point out that the debater was Roger Barker.
Yes,
you will hear from shortly.
But what really struck me at the end of that was that here was a bit of a schism opening up, a bit like in the Alzheimer's world, which I've been reading about. I've been trying to understand the amyloid versus tau arguments there. And I mean, as I say, I'm on this wonderful trial, which is brilliantly innovative, but it seems to be that there seems to be a lot more excitement about the trial and its mechanics than the actual drugs being tested.
Yeah,
which is
and it's true, and it's because one thing that trialists can fix is trial design, and so that's the bit that it's fixed. So fast. We've gone from slow to fail. Well, arguably fast to fail now, but really we want to get to faster success. And the things that sort of tip whether it will fail or be successful in finding a disease-modifying therapy still comes a lot from the biology that everybody here is doing. So, I mean, you identify a really strong gap and problem that we definitely have in the field, that these trials are exciting, but where and when.
And I'm interested in understanding where I've been into Demis Hassabis' spin-off company, Isomorphic Labs, which is, you know, incredibly impressive, and got so much brain power in you know, a fast filling up office in King's Cross. But one is struck by the fact that Parkinson's is probably quite a long way down their list. The reason being, they don't say this, but you think, oh, it's bloody difficult. There must be easier stuff to go for. So I'm beginning to feel that the AI claims in that area are possibly overhyped.
Is that the answer, do you think, AI? You have more experience, I think, with your conversations.
Oh, come on. I mean, from what I understand, all of you are using AI. We're all using AI in all sorts of different ways. I mean, just the way you're handling vast amounts of data. I mean, I'm using an app, for instance, from a company called Kneue, Kneue, which is working, I think, from Michelle Hu's data in Oxford, which measures, it's designed to be a remote monitoring app, and I have to do an exercise routine three times a week, saying R into it, holding it up, walking with it, and so on. And they are producing a vast amount of data which they're interpreting using AI techniques. But it's become like electricity. It's, you know, of course you're using AI, but is it really that great leap forward that you say it is? I mean, I can see why it might get you potentially. The way I understand it, you're walking into a forest with 1,000 trees and trying to choose the right few trees to try out, and an AI may help you towards that rather faster. But then you've got the whole clinical trials process, and we know just how lengthy and complicated that can be.
Yeah. You've been very ready to try tech out as well, which is obviously very useful. And you've heard today a few talks about different ways that people might use wearables, watches.
I've worn that wearable that we heard about.
Activity?
On the back,
Yeah.
Which is, because I was kind of disappointed when I heard originally that what the technique was for deciding how well the drug had done, and then I was impressed by the sub-study using an objective monitor, which promised potentially a bit more clarity.
So having objective measures is good, it sounds like, but we're still some way off.
The whole area of measurement is just frightening. How difficult it is. I mean, it's really come to mind. You know, when I had a tumour behind my eye, they measured it. You know, they could say it's 3 millimeters or whatever.
Yes.
That they can't measure my Parkinson's.
Yeah. Thank you.
And that is why it's. I presume that's why one of the reasons why it's so difficult.
Yes, amongst the many others.
Yes.
And do you have some advice for researchers? Apart from hurry up. But you can say hurry up as well.
What I always say is bloody well hurry up. But I mean, collaboration is obviously the key. I think it's getting better, but one hears tales of people, you know, desperately guarding their own patch. I think getting the tone right is important. Being realistic with people, because I go along to these Cure Parkinson's research updates that happen every 6 months, and they're brilliant in many ways, but there's always a moment when somebody in the audience says, "But how long?" and it all goes hazy then. And the charities, for obvious reasons, say this is more exciting than we've ever seen before. I mean, the two big charities that sort of, well, it's obviously Michael J. Fox, but Cure Parkinson's and Parkinson's UK. In the UK, their constant tune is, these are really exciting days, there's great progress being made. And to be honest, it's a bit hard for me to see that is accurate. But it's a terrible line to walk. I can completely understand, between, you know, giving people some hope and a bit of realism.
So you've also been a really big voice through the Movers and Shakers podcast and the Parky Charter. And I wondered if you can tell everyone, what are you trying to achieve with the Movers and Shakers podcast? And
well, to be honest, we started with me meeting a judge in a pub. And that judge saying to me the second time we met, should I bring Jeremy Paxman next time? And me going, ooh, and then...
Did you not know Jeremy before that?
Well, I did, but only to sort of genuflect in front of him in busy corridors. I am not worthy. But he's a lovely man. And it was the judge that sort of persuaded us to do a podcast, having first suggested a book when we were just four of us, and he said we could call it Four Men in a Boat. And Jeremy said that rightly, that's the worst idea anybody's had ever, which you have to say to the judge from time to time, because he does have some good ideas, but others need squashing. But he was right about a podcast, and we just started just recording basically in the pub our sort of inconclusive ramblings, and our brilliant producer managed to edit it into something listenable to. And we didn't have a mission except to just communicate. We hadn't heard people talking about Parkinson's, and we wanted it to be talked about. But we very quickly got this reaction, which told us that it was important, and that Parkinson's care in this country was severely lacking in many areas. I mean, the major one being the wait to see a neurologist in the first place, and the wait between appointments, and terrible stories of people basically being forced down the private medicine route because they'd been told they'd have to wait 18 months for a diagnosis. Yeah. So that formed the basis. That's number one on our list of demands, as it were, which we took to Downing Street, and we had a sing-song outside Parliament, and we had a petition which reached 100,000 signatures, which gave us a debate in Parliament. And we feel we've done well on the awareness front, but could do better on actually changing things.
Yeah, and I think that's a question that we all have. I mean, how to affect change at a political level.
The trouble is, Parkinson's got a slight image problem. Cancer, dare I say it, is exciting and terrifying. Somebody's had it, and it's I don't blame people for preferring to give money to something dramatic than something which is, shambling old men spilling their beer on the way back from the bar, in the cliche.
Not just that, but yes.
Exactly, but that's the image.
Okay, so I'm going to open it up in a few moments. Actually, maybe now. Does anyone want to ask a question before I...
Don't be shy.
Yeah. Fantastic. We've got some microphones, thanks.
Thank you for speaking and being so honest. I think this has really been really helpful and really important to have your voice as well as part of this meeting. And something that struck me that I wanted to ask what you think we should do was your comment about how the charities, but I think this also applies to how we talk in clinic, that we keep saying it's so exciting, there's so much happening, but actually is it really happening, and how is that filtering down and affecting individual people? So how should we do it? How should we be communicating, and how should the charities be communicating to balance that?
Everyone who's got Parkinson's sighs every now and then when their email inbox fills up for something their relatives have sent them saying, "Oh my God, there's this great new breakthrough." And I remember there was one. It was a story about DBS. It was adaptive DBS, but the person they interviewed hadn't had adaptive DBS, and it was presented as a radical new surgery. All one's relatives were saying, "You've got to get this." Yes, I know about it. I won't pretend it's easy, because you do want people to have some hope, obviously, but I think you've got to choose what to shout about, and also make it clear how soon that is coming in. I mean, adaptive DBS does strike me as a good story, as long as it's not presented as a cure, but as a decent story. It's the trouble is the combination of dare I say it, overexcited scientists and journalists who don't quite understand what's going on is quite dangerous. Roger.
Yeah. You could tell us about the debate, Roger.
So I was just going to say, I mean, that is a bit of a problem in the world of journalism, because you can't have the BBC headline saying interesting new idea in DBS might have some traction in the future.
Yes, no, you're absolutely right.
So it's going to have to be miraculous cure breakthrough. So how much do you think the journalists or the press have a responsibility to be more responsible with the headlines?
I completely agree. We've done at least we've done episodes on how it's covered in the media, and we actually ended up, this poor woman, we quizzed a BBC journalist about why she'd overwritten her story, which I felt slightly guilty of, because I thought I'm almost certain I did this. I won't pretend it's easy, but you're absolutely right. It's as much the responsibility of journalists as the scientists.
This is Nick. This is my husband.
Thanks, Rory. So one of the things we've been struggling with here, and we've heard a bit of it today, is getting patients involved in research, which clearly is a good thing, but also in the research planning. And one of the things I've struggled with and not solved is how can we engage with a diverse population for all sorts of ways and get them involved in helping set up the research, think about what the priorities might be. I'm thinking about the trial here, I'm thinking about other things we're trying to do with the MDC. Do you have any views on a way that we can find a way through? Because as you know from your own experience with Parkinson's, you'll meet a whole bunch of people with very strong views of all flavours, and having random noise coming in as the advisory panel for your research isn't helpful either.
Yeah. When we were campaigning for our petition, we suddenly realised that the most effective people were local Parkinson's groups. Now the trouble is they are much more active in the south. Sussex and Dorset were the top places, but there are lots of brilliant people in the Parkinson's community who will do whatever you ask to. We're running a campaign at the moment about voice as an under-recognised symptom, and the campaign has almost been taken over by half a dozen people who are volunteers in local groups, and I think it's worth getting together with Parkinson's UK and getting connections with those groups, because they are, I mean, they're the people who show you that apathy may be a symptom of Parkinson's, but it doesn't apply to everybody.
Before we do finish, though, we do have to ask you about Sophie. And should everyone have a Sophie? So tell us. I think there's a picture. Oh, there we go.
Yeah. So
what happened with Sophie?
I about 3.5 years ago, I was just finishing a book about my mother, my rather unusual childhood, and we got a dog from Romania, a rescue dog. We'd had a dog which had died, and we'd been very upset about it, and my wife wasn't ready to get a new dog, and then suddenly we were, and we ended up getting a dog from a Romanian charity which arrived in the middle of the night just before Christmas, and was the shyest dog on earth, was absolutely terrified, and hid behind our sofa for 6 months. Anyway, because I was very active on social media in those days, I still am, but not on Twitter, I tweeted about it, and very quickly the dog went viral under the hashtag Sophie from Romania. I knew it needed a hashtag, you see. And actually, this was just before Christmas, and within days, a Radio 1 DJ, Nick Grimshaw, was tweeting, "I can't believe everyone isn't following the story of Sophie from Romania." And then a few weeks later, as I was finishing this book, my agent rang me and said, I've got 5 major publishers wanting to know whether you're going to write a book about the dog. And I thought, oh God, I suppose I better write a book about the dog. So I did, and the dog has actually been a part of part of the book. Do buy that and my previous book, Ruskin Park, by the way. But the book, there's quite a lot about Parkinson's in the book, because psychologically, the dog was an enormous worry, and I used to lay awake worrying about her. But it was a distraction from worrying about myself. So in a way, she's been on the journey with me, and she's quite understanding when it takes me 10 minutes to put her harness on, which has to be done because she escapes from time to time, but she's quite patient, and it's kind of, here you go again, oh. Because my wife, who's a Cambridge professor, is away in Cambridge half the week, and my wife is a very efficient person, and the dog looks up to her in a way she doesn't look up to me. So the relationship with the dog has been quite important in my Parkinson's.
Do you have a name for your upcoming book and a timeline for it?
Well, I tried out AI and it was hopeless at coming up with titles.
Which LLM were you using?
Well, I'm using Claude quite a lot, but at the moment, working title is The Parkinson's Puzzle. Okay. Can AI decode it? But it's a bit crap, but you know, it won't be out till next year. So we've got time to think.
Okay. Well, we're going to wrap up, and I just really want to thank you, Rory, because you know your openness and your insights and telling us about your identity and your resilience and the role of Sophie in all of that is really enormously valuable for us, because I think we also get slightly lost in progress being measured in breakthroughs, whereas actually progress is measured in meaningful solutions and an impact for people. So that was really wonderful for us, and I hope you'll all join me in thanking Rory for joining us.
Thank you.
Okay, whilst we're getting set up for the final session of topics in what has been, I'm sure you'll all agree, an amazing day with some inspirational talks, we have 2 final speakers who, again, need no introduction, and I'm very excited to hear what each of them have to say. First up, we have Professor Tom Warner, colleague from Queen Square, who is the director for the Rita Lila Weston Institute for Neurological Studies, I think that's right, and the Queen Square Brain Bank. And running on a theme of today, Tom is also going to be talking about AI and big data approaches to neurodegeneration. Thank you very much, Tom.
Thank you. And thanks, Rory, for giving me the new title for my talk. Can AI tackle Parkinson's disease? It's a bit crap, really. If only I'd heard you before I'd done my slides. But you've summed the talk up very well. Right, so a bit of a spoiler alert. If you're expecting earth-shattering data, you can leave now. This is more an introduction to a program which we've started about a year ago, and we'll be running, we hope, for many more years, and we're getting up to steam. But to give you an idea of what we're doing and how we're trying to use large data sets to look with AI at things in a different way, possibly even looking for unknown unknowns. So there we go. So yes, this is the modern pathology we do nowadays. So this is we all know about neurodegeneration and neuropathology and the link between the two for hundreds of years. And the value that they've given us in terms of clinical pathological correlations, diagnostic precision. Who can forget 2004, the Queen Square Brain Bank Parkinson's diagnostic criteria? That took it to above 70% of clinicians getting it right. Pathogenic mechanisms. Pathology's played a huge role in working with the aggregated proteins across a range of neurodegeneration. They've been used as the foil for large-scale genetic studies, looking for causative genes or variants that may affect the risk of getting Parkinson's and so on, developing biomarkers, and even on occasions therapeutic targets. So those sort of studies, I like to refer them as, they're the sort of monochrome pictures. There are a few people in this audience who've heard me give exactly the same talk about 6 hours ago, so I'm sorry. The jokes are exactly the same. And what I want to take, this is the hubris bit, after which I'll calm down, Rory, is what we like to think we're now moving into an area where it's in colour, it's 3D, and it's moving pictures. And what we're doing is we're developing a large platform for interrogating large data sets, which you'll hear about, based on the brain tissue from the Queen Square Brain Bank. And we want to try and get these models to look at, identify, quantify across multi-dimensional data sets. And these are the promises you always get in these slides. So we hope to actually be able to predict clinical and neuropathological phenotype and progression. We're hoping to develop in silico models of Parkinson's and its progression, Alzheimer's and its progression in that order. We may identify some more meaningful disease subtypes. We may look for more disease mechanisms and particularly biomarkers, which will be valuable, hopefully, in the trials. And the holy grail is therapeutic targets. So the resource is the brain bank, which actually we had Rory and his team round just a couple of weeks around. Podcast coming this month, I've been told. And we are lucky to be the custodian of one of the largest and oldest brain banks of human nervous system tissue. We're approaching 3,000 cases now. It started off in just Parkinson's, and that's probably still the bread and butter, and the Parkinsonisms, moving into what are referred to as high-value dementia cases. They're all high-value, but these are ones particularly which are genetic, familial, young onset, and rare presentations. And we also cover other ones, FTD, aging controls, which we all are. And then new cohorts, we're looking now into particularly those who have been involved in disease-modifying trials, like some of them from the HD genes. Interestingly, we've got a young adult Down syndrome cohort in London who obviously get the very advanced and aggressive Alzheimer's type phenotype we're recruiting from. And perhaps we risk chancing our arm by going into CTE in a cohort of sportsmen who think they have it. We're lucky that we have access with the NHS to their entire life course data, their clinical data, which it all comes in. We've got boxes which have all gone off to be digitised just in the last few hours. And the other thing which is digitised, thanks to Zane, our neuropathologist, is the neuropathology. So we have a large open access collection now of probably approaching 1,000 cases of their pathology. And she's also developed algorithms for quantifying the misfolded proteins in all the sections. So gone are the old days where the pathology would go 1+, 2+, 3+ in terms of severity. We can actually quantify them now in the different regions. Thanks to GP2, we have whole genome sequencing on a lot of them. And then there's a lot of in-life data, which we have access to as well. And so what we're talking about is this new program. And it was we worked with the Reese Lyle Weston trustees, and they actually came up with the BRAIN, which is biomarker research and innovation in neurodegeneration. That's the BRAIN. And this is a large and ambitious project of creating large data sets across a number of hubs, which we will then try and interrogate with various forms of modeling and AI. So we have the clinical phenotype in terms of the data, we've got the records, we have the genomics, and we're building the quantitative digital pathology. We will, as part of this, be going into looking at proteomics, lipidomics, and metabolomics. An interesting trial Patrick Cullinane has been developing, looking at post-translational modifications with nanopore sequencing. And towards the end of this first 3-year iteration, we'll be doing some spatial transcriptomics. And we're doing this on initially, so half the set, so about 1,500 cases for the ones on the left, and above that, the multimodal ones, we'll be looking at smaller groups. And then we'll be adopting a array of models to actually try and identify biomarkers and look for patterns. And this is working across a number of hubs, so it's at the Brain Bank for the clinical and the pathology. We're working with the mass spectrometry unit in ICH, and then UCL Centre for Computing Science Studies with Danny Alexander and Professor Andre Altum, who was working on their team. And this is something which kicked off in earnest, probably. We've really been collecting data for the last 6 months, and these are just the staff that's been appointed over the first 6 months working in the various data hubs. So, clinical one. We've got the one busy slide on what we're doing with clinical data. And I've already got rid of it. So this is a busy slide, but in the bottom left, we're looking at the records. So what we want to do, we can't have a human going through the hundreds of thousands of pages of records, but we can use the humans in a slightly predatory way to get them to transcribe 1,000 pages, which a couple of them did. So we use that as what's referred to, I've recently learned, it's the ground truth. And then from that, we looked at around 14 different models that are off the shelf to see which on just typed pages can actually produce somewhere near the ground truth. And we've now got 3 we want to move forward with. Some of them are absolute pants and completely hallucinate whatever is on the page that they've been copied on. And others are extremely productive. On top of that, we've got some approaches now where we're going to try and de-identify them, because all our records sit in the data safe haven, which is so safe you can do no useful research on them, but no one will ever get in there. And so if we can de-identify them, we'll be able to release them to the world, and then we've got something we can actually work on in these models. So that's been working on for a while now, and we're making progress on that. And then the other 2 steps, one which we started with is clinical feature annotation. So this is, again, creating some ground truth for collecting just basic clinical data. So what we hope to do in the future, we'll have algorithms doing this. We could have an individual's every single blood pressure recording in their entire life, if that's useful. I mean, certainly for vascular risk factors, all the drugs, things that you can't do even with a well-trained research fellow for 4 years. It's something that will actually look at the scale of these data sets and draw them together so that you can then actually start doing some modelling on them. And then also we'll be moving into, this is just the typed ones. Lord help us, when we get into the handwritten general practitioner's notes, we'll be having some fun with that. And also other formats. So when someone photocopies the 2 pages in A3 format, and you've got to teach the algorithms to split them and treat them as real pages, divide them up. So every step is painful, but once we've got them taught properly, they'll be able to plough through these much faster than we'd ever hoped to be able to do it. Neuropathology. So as we were saying, we've already got quite a large digital neuropathology. So for most of the cases, there are up to 15 anatomical sites, and then they've been stained. This is a basic panel of stains looking across the abnormal proteins into inflammation and just basic structure. And those are the sort of cases we've digitized so far as part of this. If you really want, they are open access. If you apply for them, it's on the website. And these will also have the quantitative protein loads done on all these cases. So we begin to build up large data sets according to the abnormal proteins. We're also looking at cell loss, atrophy, and so we're looking at segmentation of various brain areas. We can see frontal and hippocampus, midbrain. The picture on each one denotes the member of staff who has dedicated a large chunk of their adult life into this, which we're very grateful. And this is manual segmentation of the areas because we want to compare white matter with grey matter, look for atrophy. And then one of the groups working at computing science, this is where they've started to do some modelling. So on the far left is just the basic H&E. The manual labelling is done on the next one, and then a rather standard model is trying to do it. And then the foundation model, which has been developed on this one slice, shows that it's pretty close to what the manual will do. And this is what we're hoping. If we can recapitulate that across all the sections of all the cases, this is going to create a sort of incredibly valuable large data set. In addition, we're looking at the protein loads. So we're looking at the immunostaining for the various ones and quantifying these. And keeping it moving, we're looking at inflammatory processes. So there are already some work done on T cells in both the amygdala and the midbrain. And there's one paper with Patrick and Zane, which is due to come out fairly soon. Because there's a huge interest, obviously, in neuroinflammation in most of the neurodegenerative conditions. And again, looking at the notes, we can pick up all the cases who've had systemic neuroinflammatory diseases to see whether some alteration in their immune function may predispose you to developing inflammatory brain disease. And then we're looking at vascular pathology. So gone are the days of seeing a little UBO. This is quantifying in the white matter all the small vessel disease across the range of neurodegenerative ones, because the theme of co-pathology, not just co-pathology of aggregating proteins, sort of like Alzheimer's with tau and amyloid, and Parkinson's dementia with Alzheimer's pathology plus synuclein. There is the inflammatory components, and also the vascular components which may be playing a role. And these are the things we want to factor in to these analyses downstream. And so with foundation training, this is looking at plaque burden for beta amyloid. So I think they did testing on about 80,000 slide images, and this was not done manually. And then we're left with the two things on the slide. So the upper right is the model which picks out the plaque burden for the tissue of someone clearly who has a heavy beta amyloid burden, and fortunately didn't pick it up in the control slide. Again, these are what we will then want to scale up across our entire collection. So what about omics? Well, we're working with the mass spectrometry unit, and we spent 6 months actually looking at what we do before we started. And most important one is we want to be looking at the metabolome, the lipidome, and the proteome. Is there one way of extracting that? And so there's about 3 months' work of looking at the various extraction techniques. And interestingly, one of the old-fashioned and common ones does pretty well for all three, so acetone. So this is going to save us a lot of time doing the extractions. And then they did a more interesting experiment, which is, this is new and quite interesting data, not that we can't always explain very well. Looking at, we're aware that when you're looking at the RNA in brain, post-mortem interval plays some role, although it's not as important as we thought it used to be. But we also looked in this, this is Kevin Mills's group in ICH, looked at how long they've been stored at -80 and to see if there was any effect. Proteins, there was a bit of variability. The standout one, and spoiler alert for those who've got lipidome papers out, is for some particular lipids, there was a 14 to 80% increase of that lipid per year of storage. Now, the big surprise, we thought, well, everything degrades with time. Why is it going up? And I think what we think at the moment is we may think if you stick something at -80, everything stops. Clearly, there's some process that might still go on in what cells we have, and it's this class of lipids. So the lysolipid species, this is the one, it's the middle panel 3, which seems to go up. It may be that some of them are being having some residues lopped off them to create a lysolipid, and that pathway goes on, but the next pathway, which would normally metabolize those, isn't, and we're just building these up. So I think what it's shown is, more than ever, when we do these experiments, we really have to match the controls as best we can, not just in sort of, gender, race, etc., but PMI interval, and more importantly, how long they've been stored. And in a brain bank, it's been only 40 years. That's a significant thing. But we are now ready to plough ahead with this particular data hub. So I think once we've got over all these talks, we'll actually do some work and start sampling like crazy and seeing if we can make the mass spectrometer last more than about a month. Finally, there's an interesting work from Patrick, who's been doing this. So nanopore sequencing, we're all aware of it for sequencing DNA. This is a more than one way where you can actually sequence peptides. And it's a way of identifying the amino acids. So you can predict, you can have a strand, you can have a molecule of alpha-synuclein, whatever you want, and you have an artificial pore which you create in a membrane. This is in just a tiny little desk box which just sits next to your computer. And every time an amino acid passes through the pore, it gives off an electrical charge, and that charge is enough to fingerprint it. What we're particularly interested, because we know the sequence in most of these things, is actually what it can detect post-translational modifications across the board, very much from phosphorylation to ubiquitation, glycation, all the things that we think affect at certain features, affect aggregation, stickiness of the proteins. So the bottom is, you'll all recognize a slightly unfurled alpha-synuclein molecule in all the sites where there is post-translational modification. And actually, Patrick and Mariam, who's on the far right, are working with the mass spec people, so Tatiana and Sarah in Kevin's lab have got to stage 3 of this pretty quickly, where they're actually now immunoprecipitating proteins, purifying, then trying to look at brain-derived alpha-synuclein, particularly those with disease, and comparing with controls. But still some way to go, but they've gone great guns with this particular technique. And lastly, talk about something we haven't done. This is coming up in the next year. We want to start a hub, probably in conjunction with Karen Duff's group at the DRI, in terms of spatial transcriptomics, looking at particular regions and this will be on a much smaller scale. It's not across 1,500 cases, but and already they've had some interesting findings in PSP, looking at the subcortical white matter, which I think may be the future publication. But this is another foil to the huge data sets we want to create to really allow our analysis to be as powerful as possible. So you're pleased to know, the last slide doesn't say this is great, but it's still crap. But this is where it's a fairly unique AI-driven platform. I think lots of brain banks have done some really sterling work. There's a really interesting paper recently on US brain banks collaborating together to look at PSP. They've harmonized their methods, which is something we need to do globally. We're lucky because of the country in that we have the access to all the records and also to large cohorts which have been followed up for ages, which allow us to collect and curate their brains and do the work on them. And the key final feature I want to say about this, the list is there of the aspirational goals, which we hope will come from this, is that this is going to be an open access resource. So this is not what we would ideally think. If we can get a platform that's working reasonably well and can predict perhaps what some factors might affect progression in Parkinson's, Alzheimer's, FTD, so on, or something that suggests how severe the pathology is going to be, what we really are after is people end up saying, well, can we enter our data set? We've done this on some MSA cases. We've got a whole load of Western blot data about a variety of proteins. Put that into the model. Does it alter the output? They get to know what it's done on our output. Our machine learns more and gets better. And this is something that we hope will actually be a less than normal selfish scientist sort of global resource which will benefit everyone. So one of the other things we're doing, we're federating with a lot of brain banks, particularly Sonia, who's in a number of multinational collaborative groups. So, the neuropathologists are quite good. Obviously we know their attention to detail, but actually their attention to detail and how they process and how they store the samples is pretty homonymous across the world, which is good. And also supporting the brain banks which are starting up in other countries where we know almost nothing. So the work on getting different ethnicities. As we know, most of Parkinson's data is based on middle-class white cohorts. We know that we need to be looking at other ones. So the supported brain bank, I think, is one in Nigeria, Singapore, South Africa, and Brazil. They will be really interesting to get involved in this sort of studies if we can offer them free access to look at some of their cohorts. So I told you, no data analysis. Come back in 2 years and we might have achieved something. I'm not bearing in mind Rory's warnings. I'm not going to fill you full of hubris, but we're doing something in a different way. We're doing it with more data sets than have been done before. We think we've got the right people to do it and the right technology, and we are quite excited about what potential outputs we've got, but I won't put a time on it. Thank you.
So thank you, Tom. We have time for one question. If there's a question yes.
Hello. I just want to say I really enjoyed that. I wanted to ask, sort of, what do you see the main obstacles, apart from like refining the results? What are the main obstacles you see in terms of deploying this to other brain banks, sort of internationally, and potentially being able to scale this these pipelines up to be like high throughput and things like that?
I think we can just for our 3,000 we can scale that up reasonably. But a lot of it is actually is data style. Run any one algorithm to sort of annotate the cortex. Depending on what program you use, it can take 10 seconds to 15 minutes. And also the size of data, we've had to buy a couple of terabytes to keep us going for the. So there's that, and obviously we're heating the sky at the same time. But I think in terms of federation, there seems to be, certainly amongst brain banks, a realization that if we don't work together, we are, it's a bit criminal, really. The problem with science, obviously, we all like to get our publications out first, but actually there are an increasing number of pathology, and like the genetics ones now across GP2, of telephone directories of authors, but they've all pulled together, and the data, they've managed to, GP2's got all the samples in, and people get recognised, although you'll never find yourself in there. But I think the nature of science and collaboration is becoming clearer, that you have to, for some of these things, you have to go big. For some, you still get great science from a few cells from a few patients, but this is the one where we have to work together.
Thank you very much, Tom. And last but by no means least, we have Professor Roger Barker. Roger is Professor of Clinical Neuroscience from the University of Cambridge and a consultant neurologist at Edinburgh, and really one of the pioneers when it comes to gene and cell-based therapies for a range of neurodegenerative diseases. And today's going to be talking more about Parkinson's. Thank you very much.
Thank you very much. Well, thank you very much for inviting me. So I'm going to talk about cell-based therapies. This is, it's obviously a bias because people, when they come to clinic, it's very rare to get through a clinic without someone asking me about this. Having been at the World Parkinson's Congress last week, this was a question which was asked of me endless on the roundtable discussion. 4 of the people of the 16 round the table were being recruited to the Blue Rock study. So people are entering these trials now. So this is probably a question you may well be asked. So these are my disclosures. I talk to lots of people about anything they want, including cell-based therapies. So I'm going to start about the therapeutic challenge. All of this is incredibly familiar to you, but one of the important things about a therapeutic challenge is when you start, you're starting from an academic base, but at some point you're going to have to interact with either a spin-out or a company. And so what the company will want to know in order to deliver this into the clinic may be very different to what you want to ask. And one of the sort of stories I want to take you through this afternoon is how this began very much in the world of academia. It is now very heavily in the world of pharma, and it is running at a speed which may be slightly ahead of where the data is, and I have absolutely no capacity to influence that. And that may be a good thing, coming back to Rory's point about moving things fast, but it may also be a bad thing because there are issues which may be of more importance than the trials necessarily have highlighted. So what are important? So the first thing is it's common. So that's good for a company. They like common disorders. Typical age of onset is about 70. This is likely to double the number of people in the years to come. So this is a growing problem. Companies like that. Most cases are idiopathic. So that means there's sort of an obvious genetic target, an antisense or something. You could go after synuclein, but that means that there is potential if you have some therapy that targets one aspect which is common to all people with Parkinson's, which is the loss of dopamine. And so we know that if you give people dopamine drugs, it actually prolongs life. So this is not disease modifying in the sense that it deals with the pathology, but if you had Parkinson's in the 1950s, your life expectancy would be about 10 years. At the community level, your life expectancy with Parkinson's is normal. That's due to dopamine drugs. So dopamine is a good thing for Parkinson's. It costs the economy a lot of money. The challenge will be there are a lot of good dopamine treatments out there. And so what I want to do is I want to take a novel dopamine cell therapy into people with Parkinson's. And the rationale is very straightforward, as you all know. It's also doable because in your midbrain you have about 500,000 dopamine cells, and when you lose about half of those you get the first clinical motor features of Parkinson's. So if I can put back 250,000 dopamine cells of the type that are lost in Parkinson's, so the nigral A9 neurons, to all intents and purposes I should be able to repair that part of the brain and put it back to normal. Now, why is that node important in Parkinson's? Why? Because we've all heard about these co-pathologies, pathologies in other sites. Because, of course, the treatments we use in 2026 essentially all revolve around the dopamine system. We manipulate the dopamine systems in many different ways, and then we do a whole series of further manipulations to deal with the problems that we've generated from the dopamine medication we have. That's not to take away from the non-motor and lots of the other treatments are out there, but the mainstay in clinic is around the dopamine system. So there is a very good rationale for going after a dopamine cell therapy. Dopamine drugs work. We know that's a critical node, and we know it changes people's quality of life and life expectancy. So what would be the advantage of doing this, given I've already said we've got 101 different ways of putting dopamine back into the Parkinsonian brain? Well, one is if you put dopamine cells back to the site of greatest loss, which is in the putamen, you will avoid all the off-target problems of dopamine drugs. So you'll avoid all of the neuropsychiatric problems with dopamine agonists. You'll avoid some of the exacerbations you get of other aspects of Parkinson's disease, like the autonomic problems, by not using the dopamine drugs having off-target effects. If you put back dopamine in a synaptic fashion with neurons, with physiological release, you won't get L-DOPA-induced dyskinesias. And thirdly, it's a one-off treatment. So the vision for this treatment is it will never be curative. You are not dealing with the underlying problem. But nevertheless, it could transform the therapeutic landscape from that to that. And in essence, if you could get that to work, that would be a major change with what you would see with people with Parkinson's. So the question is, how close have we got to that vision? And what I'm going to do is I'm going to take you through two source of cells. I'm going to give you the historical data around the first source of dopamine cells which were used, which were human fetal dopamine cells, and then I'm going to talk about the recent trials around stem cell-derived dopamine cells. So just to get you onto the same page, what I'm talking about here is I'm going to transplant into the brain of people with Parkinson's developing dopamine cells, and the only dopamine cells which have shown reliable results and which were really worked in the preclinical space were developed, if you like, in the 1980s, and they were taking the developing dopamine cells from aborted fetuses. So this is a seriously ethically contentious area. In the UK, as with most places, they're very tightly regulated, the work on this. In other parts of the world, this research is not allowed. It's highly controlled. As you know, in the States at the moment, they're not allowed to use human fetal tissue for any form of research which is NIH funded. So this carries quite a lot of ethical baggage with it, and that can influence how some people have interpreted the data. But what I want to take you through now is what has been shown by putting human fetal dopamine cells into the brains of people with Parkinson's. And when this work, which was pioneered by Anders Björklund and Ulla Lindvall in Sweden, began in the 1980s, you would think the conclusion would be obvious. So what I'm going to take is I'm going to take dead fetal midbrain, where the developing dopamine cells are. I'm going to dissociate it, and I'm going to inject it into the striatum of an aged person with Parkinson's. The outcome would be that would just die. Why on earth would those cells survive and do anything? It seems like a crazy experiment. And Anders Björklund spent, I think, 6 years getting this to work in the rat. It was a side project. They were actually started the work initially because they wanted to look at dopamine cell development and thought this was a clever way of doing it. You transplant in the developing dopamine cells, you watch them develop in the brain, so you can study development in an organoid, which in that case was a brain. And then they discovered they could really get it to work. And then with the adrenal transplants, the story goes on. So what has been shown by using the developing human dopamine cells from the midbrain? Well, I'm going to go through the positives to show you what you can do, and then I'm going to run through some of the negatives, all of which I think can be solved. But they are there. And if I was giving this to an International Movement Disorder Society meeting, most of the audience would think that this is an absolutely foolhardy approach that we're going down. So this has been a very controversial area, and I've certainly met a number of opponents en route. So the first is that if you take this approach of transplanting in dissociated fetal midbrain, the long-term clinical benefits can be very striking. So this is two patients that Tom Foltynie here wrote up who came from Sweden Square. They went to Sweden and had these transplants of these fetal dopamine cells. And these are the graphs. So at time 0 on the x-axis, the patients had had Parkinson's disease for 10 years. So not surprisingly, their UPDRS was about 40 or 30. So they had moderate Parkinson's. They then had 2 transplants done sequentially because of the difficulty in getting enough tissue. And it took about 3 years to see the most marked clinical benefit, even a bit longer. And that's going to become important later on when you talk about how long it takes for these to have an effect. Well, what was that effect? Well, the net benefit of this was that their UPDRS score dropped to about 20, which is about what you'd have when you presented. It then remained at that level for the next 15 years. So these people are now 30 years into their Parkinson's, 20 years post-transplantation. On their dopamine imaging, it's normal, and they stopped all their medication. So that is a pretty significant benefit. It's also associated with improvements in quality of life. So the Swedish, these were 2 of 17 patients they did in Sweden. When they looked at the whole cohort, there was a definite improvement in quality of life, not just an improvement on their clinical measures. So there are long-term clinical benefits. People improve around non-motor aspects because they don't need to take their medication, they sleep better, they move better, their cognition improves because they're on less medication. So it's also important to know that these cells don't just sit there and pump out dopamine, they are dopamine nerve cells. They make connections with the host brain, and the host brain makes connections with them, and they release dopamine in a normal fashion as much as one can assess it in an individual. So this was a beautiful paper from just under 30 years ago now, where they took a patient with a unilateral transplant. They then gave the patient amphetamine to stimulate dopamine released from the transplant, and then they looked at the receptor occupancy. So the way it would work is if you give amphetamine, you release dopamine, dopamine will compete with the D2 ligand, you'll change the binding to the receptor. And what they showed was if you look at a normal person, their occupancy drops down with amphetamine as you'd expect. In the transplant, they saw exactly the same. In other words, the transplant was releasing dopamine in the way you would imagine. This patient had a marked clinical response, and 14 years after he had that PET scan, he came to post-mortem 24 years after he'd had a transplant. He developed a Parkinson's dementia. So whilst making the point that whilst you can dramatically change aspects of the disease, you don't cure it. But this patient, a quarter of a century after the dopamine cells were put in, had a surviving transplant. So you see the picture of the brain on the left. The next one is the ungrafted side. All the brown is TH for dopamine. You don't see anything. All of that brown are cells that were put in a quarter of a century earlier with innovation of the putamen around it. So these cells survive, they innovate, they release dopamine normally, they can produce a long-term clinical benefit, and the people feel better for it. So what are the negatives? So the negatives are, as some of you will know, that there were lots of other trials that took place around the world, and we tried to do some sort of little meta-analysis of this. And essentially, what we found is if you look at the 2-year data, so this is 2-year post-transplantation, a 33% improvement in your UPDRS off medication we thought would be bigger than any placebo effect, and a third of patients who had had this procedure across multiple centres, all of which did slightly different things, had a significant clinical response. But that means 2/3 don't. So you can't take someone to theatre and say you've got a 1 in 3 chance this will help. So it's inconsistent. So if this is going to work, you're going to have to do better than that. And there may be reasons why there were inconsistent results. There was also this very curious case, which is often brought up, of a patient in the States who had a transplant, and at post-mortem, they had the most dopamine or most TH positive cells in their brain in the transplant site. They had never shown any response to the transplant, but they continued to show response to dopamine medication. I have absolutely no explanation for this, because there's no reason if you had dopamine cells in your brain they wouldn't be responding. The question was whether they were fully mature dopamine cells, and TH is only a surrogate marker for the dopamine system. But that's one case. The third, which is often cited, is these transplants acquire the pathology of Parkinson's, and we heard that earlier on this afternoon. So there were 2 cases reported back in 2008 where they showed that the transplanted dopaminergic neurons in the transplant had acquired the pathology of Parkinson's. These dopamine cells in these transplants are about 10, 12 years old, so they shouldn't have any pathology. So that opened up this whole idea of synuclein spread. It's fair to say, whilst the population of cells affected by this is real, the numbers are small. So at 24 years in that transplant, about 20% of the dopamine cells had Lewy body pathology. So at that rate of acquisition, this transplant would probably fail after about 50, 60 years, by which time the patient would have failed for some other reason, I would imagine. Then the other major problem was in the NIH studies, which were these American studies, which failed to hit their primary endpoint. So they didn't improve the patients in the endpoint they were looking for. A significant number of patients 15% in the first trial and 54% in the second trial, developed these graft-induced dyskinesias, which were unfortunately called runaway dyskinesias when they're originally described, which is not a helpful phrase because it implies once they start, they're never going to stop and get worse and worse. They were just graft-induced dyskinesias, and of course you can always get rid of L-DOPA-induced dyskinesias because you can always stop L-DOPA. This you could not stop because you've got a graft and you can't get it back. They did respond to DBS, but it was a worry. And whilst no one's got a full explanation for it, the most likely reason for this is it's the contaminating serotonin neurons which develop adjacent to the dopamine cells, which are mishandling the dopamine and driving the dyskinesias. And that is something you can solve with stem cell derived dopamine cells, as you can the pathology, because you could make alpha-synuclein knockout cells. But the other big problem when we discovered this in our final trial, which I think will be the final trial of fetal dopamine cells, the TransEuro study, was it's very difficult to get sufficient tissue. So in order to have enough dopamine cells to transplant one side of the brain, you need at least 3 human fetuses where you can dissect out the midbrain. So you've got to collect that within 5 days. So in our study, which we did, TransEuro, we ended up grafting 11 patients. So we did 21 transplants. For those 21 transplants, we had another 87 surgeries booked, which we couldn't use because we didn't have sufficient tissue. So you had a 1 in 5 chance of actually having a transplant. So that for the patient is completely unacceptable. For the hospital, it's unacceptable. As a way forward, it's just not viable. All of these, I would say, you can solve with what came next, which is the stem cell-derived dopamine cells. You can stop them getting pathology because you can alpha-synuclein knockout. You won't have 5-HT neurons. You can make them and store them in the fridge. You should be able to get more consistent data results because you can make a standard product. So this is where things suddenly changed in the field from something that was of academic interest, but changed around 2011 and 12. And up to this point, the major problem in the field of stem cells, so human embryonic stem cells came around in 98. Jamie Thompson made them. iPS cells, human iPS cells, Shinya Yamanaka in 2007. But the way to make dopamine cells that people were using up to this time was based on a mouse developmental approach, which didn't work. They then discovered, well, Lorenz Stude in particular, discovered a whole new way in which these cells developed, and that then suddenly transformed the field. And so Lorenz's group in New York and Marlin's group, Marlin Palmer's group in Lund, discovered how you could make human embryonic stem cells or human pluripotent stem cells into dopamine cells, which looked fantastic. So that's a rat which had a lesion. All of that brown is an human ES-derived dopamine cell innervating the striatum. And so over the next few years, this had to be converted from a lab-based protocol into a clinical protocol. And for anyone who wants to do that, it is incredibly boring. So the first thing is if you go for a clinical grade reagent, you just stick 3 noughts on the end of the price. Incredibly expensive. And the amount of work you need to get one paper to show you can do it is enormous, and it's normally not great for people's careers. But anyway, Agneta Kirkeby, who is very successful in Copenhagen, took this on. And it took us, 2007, 2020, it actually took us from 2013 to 2020 to make a GMP grade product which we could take into the clinic. And that has now been manufactured, and that sits currently in the freezer at the Royal Free, where we've made all of these cells. Now, for reasons that I do not understand, at the beginning of 2017, or 2013, when we were working on this work, the amount of investment in stem cell-derived dopamine cells in the world was approximately $0. By the time we started to do this trial, the investment in this was several billion. Nothing had been published on a trial. Nothing seemed to have changed. But for some reason, this caught fire. And the sort of reasons, I think, is that one, I think the shine was going off DBS. People thought it wasn't quite all it was meant to be. And secondly, the manufacturing of these cells, which is a very boring thing down there, is incredibly efficient. So if you start from 1 6-well plate of ES cells, in 16 days you can make enough cells to treat 300 people. 1,000 plates, I can treat 300,000 people. I can treat everyone in Europe and America. That's one manufacturing run. That suddenly makes a lot of economic sense if you can do that. So suddenly, the world got excited. So this is currently the situation. I think there are more companies doing it than this. Most of these people are now in trials. So Aspen Neuroscience have finished their trial. Arizona State University, they've pretty much finished their trial. Mass General, they had a single case. They've now got permission to do one trial, and Ulla Isaacson has a company that's doing another trial. Blue Rock I'll come on to talk about. There's our study, which I will talk about. In China, there are at least 3 trials, of which I've reviewed 2 of those trials. And in South Korea, they've already published, and then there's the group in Japan. So this is seriously alive. So these are the completed trials in terms of the ones which have been published. I'm not going to talk about the first one. The first one is a curious study, and I'm very happy to talk to people about that because there were interesting aspects about how that particular work was funded. But I'm going to go through each of these trials. I was going to present our trial, but I was told last night I'm not allowed to present the data from our trial because it's just been accepted for Nature Medicine. I'm told there's an embargo on the data. But if you look carefully at all of the pictures, you will get the data. It's just not in the format which will appear in the paper. So this is the first study. So this is from Blue Rock. So Lorenz, Studer and Vivian Tabar set up a company called Blue Rock. Blue Rock was then acquired by Bayer. So this is big pharma behind this. So Bayer also own Bayer, which does the GDNF trial. So this was a paper that appeared in Nature last year. So it's a phase 1 study. So they take human embryonic stem cells. So these are allogen egg. It's the H9 cell line. They use a differentiation protocol which was developed in that lab, which is very similar to the one we have used. They cryopreserved. They did all their preclinical testing with their cryopreserved product, and then they did 5 patients at a low dose and 7 patients at a higher dose. They then immunosuppressed them for a year, followed them for 18 months. They presented the 3-year data at the AD-PD meeting, which I didn't go to, but I think the 3-year data is very similar to this published data. And this is what they found. So what they found was over the 18-month period, obviously the lower you are on the graph, the better you are. So the 5 low dose and the 7 high dose, you can see that the mean UPDRS has improved in both sets of patients. Now, very important is that these cells in animals take at least 6 months to work. So if you do drug-induced rotation, standard thing, you won't see anything for 6 months. So it takes at least 6 months to see an effect. So block out the first 6 months, and you can still see that there is an effect, perhaps not quite as big as is claimed in the paper, but there is an effect. If you then look at their PET data, which is in the supplementary aspects of the publication, there isn't much on the PET data. So what you would anticipate is if you're making people better with the dopamine cell therapy, the dopamine levels in the striatum would go up, their dopamine drug requirements would go down, and that's the direction of travel. So they saw a clinical benefit. There was no change in the medication, although they were trying to keep that stable during the first 18 months, and there was no major PET signal change. So what they showed was it was safe. There were no major complications. There were clearly a dose-dependent clinical effect, but on PET imaging, you couldn't see a strong signal, which is a slight concern, and there were no change in the drugs. So on the basis of that, what would you do next? Well, you might want to think about how you're going to perhaps try and improve on your PET with different doses, but actually they were offered accelerated approval by the FDA, so they've jumped straight to a phase 3 study. So they are now doing 102 patients in their Expedite 2 trial, 2-to-1 in favour of transplant. So a third of these patients will actually have sham surgery or imitation transplant. 2/3 will have it. This trial has already begun, and as I say at the meeting I was at 2 weeks ago, there were a large number of patients who were asking me about this study and whether this was a good thing to go into. So that trial is up and running. That trial will deliver its results probably in about 2-3 years' time. If it's positive, they will get a license. If it's negative, who knows? But it's going to be difficult to come back from that to some extent. The second trial I want to talk about, which was in the same issue of Nature, was Jun Takahashi's group in Japan in Kyoto. So not surprisingly, they used IPS. And what they did was they had a master cell bank. They get their IPS cells for each patient, wake it up, and then they run it through a differentiation protocol and transplant those in. So each patient doesn't get the same cell product. It starts from the same source, but there may be slight differences. They had 3 patients at 1 dose, 4 patients at a higher dose. They used slightly less immunosuppression. They used tacrolimus. They followed patients over 2 years, and they found a significant clinical benefit in their patients, rather irrespective of the dose. If you apply my 6-month rule, then you have a slightly different interpretation of the clinical response to these transplants. Now, in contrast to the study in New York, they saw some quite clear PET imaging data in support of graft survival at some of the sites where they transplanted the tissue, but they saw no reduction in the amount of medication the patient took. So this trial, so remember now we've got 12 plus 7, so we've got 19 patients in total. This was safe. There were variable outcomes. I think you can interpret whether there was a clinical response to the transplant or not. Some evidence of graft survival on imaging, no change in the amount of drugs. So again, what would you do with that data? Well, Japan has a very different regulatory landscape. So first of all, this is now in a company called Sumitomo, who are actually taking it to a trial in America. But in Japan, if you complete a phase 1 study, you can then apply for a license. You then have 7 years to show whether that therapy works. So in Japan, this has now got a license, and you can have an iPS-derived dopamine cell transplant in Japan. I think it costs you $350,000, and for the next 7 years, you could have that treatment and see whether it helped. That's the data that underpins that trial and that license. And then the final one, which is from South Korea, theirs is slightly, you see where they all get published. They get published in Nature Cell. If you did an experiment in the lab or a primate study with this, I'm not sure you'd quite find it as easy to get it in these places. Anyway, this is in Cell. So this was a 12-patient. They've only got the 12-month data here. They used a human embryonic stem cell. They turned it into dopamine precursors. They transplanted it. What they found with their low dose and high dose was, again, there was a clinical response, but it often preceded this 6-month time point. They showed an increase in F-DOPA in the areas of transplant. I've highlighted it there. I'm going to compare it to the best patient we had in our transuro transplant study. So this was a patient who had fetal tissue who actually had a very marked clinical response. And this is what their PET scan looks like in comparison. So their dopamine levels have actually gone back to normal. And interesting, I was looking at their 10-year data yesterday, and it seems to be very sustained. So in this study, it was safe. There were some clinical effects again at the higher dose, limited graft survival on PET imaging. This is a company that's running this, so they will again, I don't quite know what the rules will be in South Korea and where they're going to take this. And then we have our own study, STEM-PD. So this was a study which we began and we staggered very slowly, so we were very cautious about this. We did it. Last patient was grafted in October of 2024, so we've now gone to 18 months in that patient. We did a lot of assessments, a lot of imaging. This illustrates how this type of work begins. So it started with our work really on the clinical side, the Lynn team on the cell side. We came together. We actually were planning to move towards a trial. We had great difficulty getting funding. The EU wouldn't fund us back in 2015 because it was too long for us to get to a trial. We need to be there in a year. We had money from the Wellcome to develop it up, but whilst I was discussing, which we were awarded, but in the time it took me to negotiate the contract with it, all the IP had been sold to Novo Nordisk, so we never got the Wellcome grant. Novo Nordisk then took it on. They funded our trial. Novo Nordisk last autumn pulled out of cell therapies completely for every indication, dopamine, islet cells and cardiac. So we suddenly were now left with a program with no funder, and Cellular Intelligence bought it or acquired it from Novo about a month ago. So we're back in business with that. What we did was we had Cambridge and Lund patients. They all went to Sweden and had the transplant. We had 2 doses. We did multiple deposits within the putamen, exactly as we'd done for the fetal tissue. I can't tell you the clinical data, but it's not vastly different from the other studies. The big complication we had was one of our patients died from a fungal infection 10 weeks after they had the transplant, which in part relates to their immunotherapy. Like the other studies, though, we have not seen any graft-induced dyskinesias, and we have not seen people go back to normal. The patient who died, we did find cells in their brain. So they definitely did have surviving cells in their brain. Now, we've done a limited meta-analysis of the trials which have been done. You can see the size of the clinical effect on the left, so it's very variable. So in our trial, we didn't see as much of a clinical effect at 12 months as other people had. What we did see was quite a marked reduction in medication. So to summarise where we are with stem cell-derived dopamine cells in Parkinson's disease, and to put it into context, you know, at the roundtable discussion last week, a lady said to me, I'm going for an interview on Thursday to have a stem cell therapy with Blue Rock Parkinson's disease. Would you do it? And I said, no, I would not. Even though I spent 35 years of my life working on this, I would not go for that trial. And I think the reason is it is feasible. It is safe. There's no problems with the product, but the immunotherapy is not without risk, but I think we can manage that. Can we put the brain back to normal in terms of dopamine levels? No. Nobody's got close to that yet. Can we make people better? We can, but we're not quite sure why. So I'm not quite sure why patients have got better. At the end of the day, if they get better and they stay better, I don't really care, but it would be nice to have a clear mechanism for that. And can it be done without side effects? Well, we haven't seen graft-induced anesia, so probably we can. So I think there are a number of key issues that we need to solve. One is we still don't know how to deliver these cells, so there's no device for doing this. We're hijacking systems to use. So that may be a problem because we may be compromising the cells when we deliver it. I think we vastly underdose people. So I think we're way out with the dose of cells. And I think that rather prosaic thing needs to be sorted out. The immunotherapy is still an issue. Ultimately, can we do this consistently with a size of response which will compete? And so finally, this was the group that put us all together. A wonderful set of colleagues, especially the team in Cambridge and my wonderful colleagues in Lund. This was the entire R&D team at Novo that helped us. Every single one of those is now out of a job. So despite the fact in academia you always worry that you don't have enough money, you do have a degree of security beyond what they had. And these are all the other people who've helped with some of the work, most of which I haven't talked about. But anyway, and with that, I will finish and thank you. Thank you very much. I think we've got time for one question.
So this is really fantastic, but if immunosuppression is the problem, so you just make autologous IPS. I think the first trial you didn't cover did that.
Yeah. So autologous has the advantage, obviously, you probably don't need to use immunosuppression, but some of these things, even autologous IPS can sometimes generate an immune response. In the first trial, what it demonstrated in the first patient was that they felt they were much better. So their PDQ-39 dropped from about 60 to 0. Their UPDRS didn't change and their PET imaging didn't change. And the patient had been quite keen on supporting the lab's work over a number of years. So the other point I was going to say, the trouble with autologous is obviously you've declared that you're not very good at keeping dopamine cells alive because you've got Parkinson's. So there is a worry with autologous that the cells will go quickly off. Yeah.
Roger, thank you. That was just a great overview. I do not know how to work this. But what I wondered was, especially now that it's regulated or the license is there in Japan, and people are coming forward to say, to you, but to other clinicians, what makes a good candidate for this? Like, do we know yet? A bit like with DBS, we have actually a pretty good sense of who is going to benefit and who won't. So where would you guide
I think they're the same. So I think all the patients who are good for DBS, pump therapies, are going to be the best patients for this. So these are generally younger patients who've got clear responses, motor fluctuations. They're the patients who would do best with this. But we don't have enough data on that. But when we did the meta-analysis, the better patients were younger, less advanced, less dyskinesias, and actually had a longer follow-up and immunosuppression.
Right, I think that's everything. Thank you so much.
Thank you.
And I think we now have a close from Tony.
Well, as it says on the slide, thank you very much. Of course, there are a number of people to thank. Most importantly, I want to thank the speakers for an amazing day, really extraordinarily inspiring, all about trying to cure as yet uncurable diseases in the most novel ways, trying to understand them and apply treatment which, in the case of Parkinson's disease, Huntington's disease, will slow and potentially prevent these diseases. So a fantastic and inspiring day. I'd like also to thank our three speakers, Murat, Sarah, and Roger, for inspiring talks, and thank you very much for coming today. I'd also like to thank all the people that put the day together, and the board, and in particular Sonia, for helping keeping us all on track and organising the day, and of course the team, and these are only a few of the team that have helped support all of the work that has been done that you've seen from Queen Square. Hannah and Angelina, here is Hannah. Angelina, I knew she wouldn't come and she's too shy, but those two have been really incredibly hardworking in putting all of this together and ensuring it goes very smoothly. And of course, there's Linda, Clement, and Imran, who also worked hard behind the scenes. So thank you to all of them. Thank you to our sponsors who have sponsored the day, sponsor our research as well. Without them, of course, today and all of the work you've seen could not happen. So we really appreciate their contributions and participation in our research. The Movement Disorders Society have supported this meeting, and this is a plug for the Movement Disorders Society meeting in Seoul. If you're not a member, you should become a member. And the membership benefits are listed there. So I think we've done our due diligence to the Movement Disorder Society for helping support this meeting. So I shall now bring the day to an end. Again, I'll say a really fantastic day. I've learnt an enormous amount and really been inspired on the breadth and depth of research across the neurodegenerative movement disorders and other movement disorders spectrum. So thank you all very much for your attention, great questions, and please do join us for a so-called networking reception, but you don't have to network, you can just eat and drink. But thank you all very much indeed.